యా సార్ లైవ్లోకి వెళ్తున్నాం సార్. యా ఎస్ సార్ స్టార్ట్ చేసినాం సార్, లైవ్ అవుతుంది.
Shall I start, sir?
Yeah, we had started, sir. Just you can go live, sir.
Okay. So, good afternoon, everyone. Myself, Satish.
Yeah.
I'm working as a senior lecturer in botany.
Yeah.
Senior lecturer in botany
Yes,
from.
sir. Yeah. Start చేసినాం సార్, లైవ్ అవుతుంది.
Start చేయాలా, sir? Okay, sir. Right. So, I'm K. Satish Kumar, senior lecturer in botany from Excellentia Junior College. So, right now in this session, I want to discuss about the botany questions. So, I'll start the botany question paper discussion. Right? So let's start with this question. I'm discussing about the code 12 question paper. So those who are following the paper discussion online, you just note down the paper code, that is code 12. So the question 92, which of the following is the site for active rRNA synthesis, ribosomal RNA synthesis? So here, nucleolus, it can be considered as the site for the active rRNA synthesis. So the nucleus, which is a double membrane bound cell organ, the nucleus mainly contains an oblate structure. We can call this structure as nucleolus. So this is nucleolus, and we can call this one as chromatin net or chromatin fiber. So this nucleolus, it can be considered as the site for ribosomal rRNA synthesis. So the answer for 92 question is option 1. Then question number 93, it's a matrix matching type question. And the topic of this question is, so they ask the several phases like G1 phase, S phase, and G2 phase. So we can call these phases as a group of interphase. They all comes under interphase. So this interphase comprises of 3 phases, G1, S, and G2. So another phase is there, that is M phase, it is also called as mitotic phase. So during G1 phase, the cell is metabolically active. So here A2, during G1 phase, cell is metabolically active and continuously grows, but does not replicate its DNA. Then B, S phase. During S phase, synthesis of DNA occurs and the amount of DNA per cell will become doubles. So that's why this S means DNA synthesis phase. DNA synthesis phase. This phase also called as auto-duplication phase, where during this phase, DNA content of a cell will become doubled without changing the chromosome number. So B suits for 3. Then G2 phase. So during G2 phase, some special proteins mainly synthesized during G2 phase, that is C4. For example, you can take one protein here, tubulin proteins. The proteins that are mainly synthesized during G2 phase, they are tubulin proteins. Next one, M phase. So M, actual cell division occurs. We can also call this phase as mitotic phase. So this M phase, for our convenience, we are dividing into 2 phases. One is the karyokinesis, which includes prophase, metaphase, anaphase, telophase. Next one is cytokinesis. So the answer for this question is A2, B3, C4, and D1. First option is the right answer. A2, B3, C4, and D1. So let's go for next question. That is, identify the correct statements about biomolecules. So this question is related to biomolecules. So I'll read the each and every statement here. Statement number 1A, lipids are generally water soluble. It's a wrong statement because lipids are water insoluble. Proteins are polypeptides. Yes, a chain of polypeptide undergoes polymerization and form proteins. That is correct statement. Polysaccharides are long chains of sugars. Yes. A single monomeric unit, that is monosaccharides, undergoes polymerization and form a polysaccharide. And these polysaccharides, again, there are two types. Number one, homopolysaccharides and heteropolysaccharides. So two types of polysaccharides will be there. One is homo and another one is heteropolysaccharides. So it is the correct statement only. Then adenine and guanine are substituted pyrimidines. Wrong statement, because adenine and guanine, they are not pyrimidines, they are purines, nitrogenous bases. So these purines, adenine, guanine, and the pyrimidines, there are three types: cytosine, thymine, and uracil. So next statement E, almost all enzymes are proteins. Yes, almost all enzymes are proteins. That is the correct statement. So the answer for 96 question is B, C, E. So second statement, second option is the correct answer. Correct? So let us see the next question. So question number 97. This question is related to photosynthesis, Calvin cycle. How many ATP and NADPH molecules are required to make one glucose molecule through the Calvin pathway? So Calvin cycle, also called as Cipri pathway, and it is the major biosynthetic pathway in all types of plants, like in Cipri plants, in C4 plants, and also in CAM plants. So it is the most common biosynthetic pathway. So here, for the formation of one glucose molecule in Calvin cycle, 18 ATP and 12 NADPH will be required. So it is the direct textbook point. So 18 ATP and 12 NADPH are required. So the answer for 97 question is option 1 is the right answer. Next one. Question number 98. Which of the following statements are not true regarding? So we need to identify the incorrect statements here. So this question is related to biotechnology principles and processes. So they are called molecular scissors. Yes, the restriction enzymes are also called as molecular scissors. Sometimes we can also consider them as chemical knives. Another name for these restriction endonucleases, they are also called as chemical knives. Because the main purpose of these restriction enzymes is they mainly recognize a particular site within the DNA sequence and cuts the DNA sequence. So that is the main purpose of this restriction endonucleases. So other name for restriction endonucleases, molecular scissors, it is correct option, correct statement. These are the enzymes responsible for restricting the growth of bacteriophages in E. coli. Yes, correct option. This one also correct statement. Because these bacteriophages, yeah, the E. coli organism, they are mainly infected by some viruses. We can call those viruses are bacteriophages. So bacteriophages, they are the viruses which mainly infect the bacterium. So to protect themselves from these viruses, the E. coli bacteria releases certain enzymes. One of that important enzyme is restriction enzyme. So B is correct. They cut the DNA only. With this word, we need to identify whether it is a true or false statement, because these restriction enzymes not only at the center of the palindromic site, they can also make some staggered cuts that produce sticky ends and also blunt ends. So two types of cuts will be there. So not only exactly at the center, they can also cut the DNA sequence away. That's why C option, it can be considered as wrong statement. They remove nucleotides only from the ends of DNA. So here, removal of nucleotides is not by endonucleases, exonucleases. So restriction enzymes are of two types. One is endonucleases and one is exonucleases. So removal of nucleotides mainly takes place by the activity of second type of restriction enzyme, that is exonuclease. So it's wrong option, wrong statement. They recognize specific palindromic base pair sequences. Right, correct answer. Correct statement is wrong. So the answer is C and D. Incorrect statements were C and D. So next question, question number 100. In which of the following the ovules are not enclosed by an ovary wall and remain exposed? So this is the question related to plant kingdom. Plant kingdom question. So Selaginella, they are the non-flowering, non-fruiting plants. So we can eliminate. It's a pteridophyte. Funaria. So it is also a cryptogamum, non-flowering plant. So it's a moss. We can eliminate this one. So then Pinus gymnosperms, which belongs to Coniferopsida. So these Pinus, it's a gymnosperm where in the gymnosperms, the ovules, they were not enclosed by an ovary wall. That's why the answer for this question is Pinus. So Wolffia is the smallest angiosperm. So in Wolffia, the ovules were enclosed within the ovary wall. So 100 question, the answer is Pinus. It's a gymnosperm. Remember this point. In gymnosperm, the ovules are naked, right? So the answer is third option. Next question, 101 question. It is related to morphology of flowering plants. Placentation. The topic is placentation. So arrangement of ovules within the ovary or placenta is called as placentation. So placentation mainly of types of placentation they mentioned here: marginal placentation, axial placentation, parietal placentation, precentral placentation, and basal placentation. So marginal placentation always found in Fabaceae members, that is P, A2. Axial placentation, China rose, tomato, and lemon. They gave lemon option, B4. Parietal placentation found in two plants, mustard and argemone, Brassicaceae members, C1. And basal placentation found in Astraceae family, that is marigold, chrysanthemum, and sunflower. So the answer for this question is A2, B4, C1, D3. Third option is the right answer. Third option is the correct answer for question number 101. Then question number 102. So in angiosperms, root hairs arise from which one of the following regions of the root? So this question also related to morphology of flowering plants. In the morphology of flowering plants, this is the question related to regions of root. So the root hairs, if you see the regions of the root. So this is a diagram of a typical root. The typical root mainly comprises of root cap zone. We can call this region as root cap region or root cap zone. So this region we can call as region of meristematic activity, region of elongation, and this is the region where the root hairs will arise. We can call this region as region of maturation. So root hairs, they mainly arise from the region of maturation. So four zones are there mainly in a typical root. Number one, root cap region, meristematic region, elongation region, and maturation region. So these root hairs, they are thin, delicate, slender epidermal cells, which are very helpful for the absorption of water and mineral salts. So the main purpose of these root hairs is absorption. So they are mainly arise from region of maturation. The answer for this question is 102. Fourth option is the right answer. Next question. Which one of the following is not a characteristic feature of plant cells in the phase of elongation? So this question is related to plant growth and development. In the plant growth and development, we are having phases of growth. The top subtopic is phases of growth. So we can eliminate the question by seeing the options. Increased vacuolation, it is the characteristic feature of elongation. Then cell enlargement, yes, due to turgidity. With the intake of water, the cell can elongate and increase in its shape. So yes, it is also one of the character of elongation. New cell wall deposition, yes, new cell wall materials also deposited during the growth of the root. Large conspicuous nuclei. So we can eliminate this option because large conspicuous nuclei mainly we can observe in meristematic region. So the answer for this question is 103. Option 2 is the right answer. Option 2 is the right answer for 103. Next one. Which of the following statements are correct with reference to transcription unit? So this question is related to molecular basis of inheritance. Within the molecular basis, the subtopic is transcription. So a transcription unit in DNA is defined by three regions. Yes, promoter, structural gene, and terminator. Correct. The promoter is said to be located towards 5' end of the structural gene. So this is C statement. The promoter is a DNA sequence that provides binding site for RNA polymerase. Correct statement. The promoter defines the template and coding strand. Correct statement. So A, B, C, D also correct. Then the promoter is located towards the 3' end of the coding strand and it defines the end of the process of transcription by releasing some termination factors. You can call them as row factors. So the answer for this question is all statements are correct here. No wrong statements will be there. So the answer is 1. Option 1 is the correct answer for 104. So we'll go for the next slide. Next one. Question number 105. Alpha helix is found in which level of protein structure? So this is a question mainly related to biomolecule structure of proteins. So protein structure is mainly classified into 4 types. Number 1, primary, secondary, tertiary, and quaternary structure of protein. So here, alpha helix and beta sheets. So 2 types are there: alpha helix, beta sheets, and one more structure also there: collagen triple helix. So these are the 3 types of secondary structure of proteins. Alpha helix and beta sheets given by Linus Pauling. Collagen triple helix discovered by Sir G. M. Ramachandran. So they are asking about the alpha helix. It can be considered as a secondary structure of protein. Other structures which can fall into this category were beta sheets and collagen triple helix. So totally there are 3 types of secondary structure of proteins are there: alpha helix, beta sheets, and collagen triple helix. So the answer for 105 is 4th option. 106 question. Which of the following statements are correct regarding amino acids? So one more question from biomolecules. They are substituted methanes, yes, with an amino group, with a carboxyl group, and with a central atom, right? Next one, serine is an aromatic amino acid. So totally 20 standard amino acids are there. So out of those 20 standard amino acids, they give 3 amino acids. So serine, it's not an aromatic, it is an alcoholic amino acid. Serine and threonine, they're alcoholic, they're not aromatic. So wrong statement. Valine is a neutral amino acid. Yes, correct statement. Lysine is an acidic amino acid. No. So lysine, it is a basic amino acid along with arginine. So the answer for this question will be A and C only. A and C only. So 106 answer, 3rd option. So next question. The main function of buliform cells in grasses. So these buliform cells, they can also called as motor cells. Another name for buliform cells, motor cells. So these buliform cells, characteristically, they mainly found in monocots, not in the dicots. So the isobilateral leaf, we can say. So a typical isobilateral leaf or a monocot leaf mainly comprises the buliform cells. So during water stress conditions, these buliform cells, they curl the leaf inwards and prevents water loss. The main function of these buliform cells is they are mainly useful to minimize water loss during transpiration process. So the answer for this question 107, it's a simple level A question. 3rd option is the correct answer. Next one, 108. So this question is related to plant physiology, photosynthesis. Find the incorrect statements about photosynthesis from the following. The water splitting complex is associated with PS1. So it is wrong statement, dear students, because the water splitting complex is associated with PS2 complex. It's not PS1. So A is wrong. C4 plants use the C4 pathway, C3 pathway of carbon dioxide fixation at the main biosynthetic pathway. Yes, the most common biosynthetic pathway in all plants, whether it is C3, C4, and CAM, it is Calvin cycle only. So B is the correct statement. 3rd statement, in C4 plant, photorespiration does not occur. Yes, C4 plants, they lack photorespiration because in C4 plants, the excess carbon dioxide, it can fix in bundle sheet cells where the Calvin cycle can be operated. So C statement is correct. C3 plants exhibit Cran's anatomy. No. So Cran's, it is a special type of anatomy where the ring-like arrangement of bundle sheet cells mainly we can observe around the vascular bundles. So the trans anatomy, it is characteristically found only in C4 plants, not in C3. It's a wrong statement. So the incorrect statements in 108 question were B and C only. Third option is the right answer, B and C. Next question. So one more match the following question. This time in botany, nearly 9 matrix matching type questions came. Okay, so 9 questions. So this is also one of the matrix matching question. So question is related to anatomy, conjunctive tissue. So conjunctive tissue is a tissue between xylem and phloem, A3. Next, Casparian strips. Those are the endodermal cells with suberin deposition, mainly found in a root. Next one, subsidiary cells, specialized cells in the vicinity of God cells, C1. Then starch sheet, it is mainly found in dicot stem. So D2, 3, 4, 1, 2 is the answer. Third option, 109, 3. 3, 4, 1, 2. Next question. So this is also one of the matrix matching type question related to both biotechnology principles and biotechnology applications, 2 chapters. So the genetically modified organism, so it is BT cotton, A2. Then thermostable DNA polymerase enzyme, B3. It is extracted from a thermophilic bacterium called as Thermus aquaticus. TA plasmid, Agrobacterium tumefaciens. It's a soil bacterium. Then PBR 322. 4 equally. So 2314 is the answer. Third option. 2314. So this is about the question number 110. It's a easy level 1 matrix matching question. So next one, 111. So this question is related to plant growth and development. Heterophyly. Heterophilous development in response to environment is example of which of the following phenomena. So then heterophyly comes under plasticity. So what is meant by plasticity? In response to the external environmental conditions, the plants can show certain morphological changes. We can call that mechanism as plasticity. So heterophyly means appearance of 2 different types of leaves. Hetero means 2, phylloids or phylus means leaves. So this heterophyly is of 2 types. One is the developmental heterophyly, another one is the environmental heterophyly. Environmental heterophyly, buttercup, ranunculus plant. Developmental, cotton, coriander, and larkspur. So the answer for 111 is it is plasticity. Next one, 112, a very easy question, that is inflorescence. So what is meant by inflorescence here? Arrangement of flowers on the floral axis is known as inflorescence. Inflorescence is of 2 types. If the flowers born in acropetal manner, it is called as racemose. If the flowers they born on basipetal manner, it is called as cymose. Basipetal, cymose, acropetal, racemose. So the answer for 112 is third option is the correct answer. 112, 3, 111, 4. Next one. So one more catch the following question. It is related to genetics. Incomplete dominance. Incomplete dominance. It is mainly observed in flower color in antennatum species, that is snapdragon A2.
Switch off patients.
Codominance. So codominance, it is mainly observed in blood grouping in animals and lentils. In lentils also we can observe the codominance. So B4. Then pleiotropy. So pleiotropy, example for pleiotropy, we can take phenylketonuria, C3. Then polygenic inheritance, human skin color. So the answer for 114, A2, B4, C3, D1. First option is the right answer. Simple question. This is also level 1. Then sexual reproduction in flowering plants question 115. Arrange the following in the correct developmental sequence related to microsporogenesis. So microsporogenesis means the formation of microspores, or we can say pollen grains from a diploid pollen mother cell. We can call it as microsporogenesis. So I'll arrange in a sequence of order. First, sporogenous tissue. It contains some diploid cells called as pollen mother cells. These pollen mother cells form a tetrad of microspores called as microspore tetrad. Then forms a pollen grain or microspore. So the answer for this question is BDAC. Third option. This is sequence. BDAC is the answer for 115. Next question. Identify the correct sequence of steps in PCR, polymerase chain reaction. It is also called a thermocycler. So the polymerase chain reaction or thermocycler, it is mainly useful for the amplification of our DNA or gene of interest to produce billion copies of DNA fragments. So the exact sequence for PCR is number one, denaturation. The double-stranded DNA denatured at certain temperature, above 90 degrees centigrade. Then annealing, joining of the primers to the ssDNA strands. Then extension with the activity of the Taq DNA polymerase enzyme. The DNA strands will extend and the cycle will continue for several generations and produces billion copies. So the answer for 119 is denaturation, annealing, extension. First option is the correct answer. Next question. This is related to biotechnology principles. Which of the following statements are correct with respect to DNA separation, isolation, and visualization? The cutting of DNA is done by molecular scissors. Yes, they are also called as restriction endonucleases, restriction enzymes, correct? The DNA fragments separated according to their size in agarose gel upon electrophoresis. Yes, correct. The separated DNA fragments can be seen without staining when exposed to UV light. No, we can't see the DNA fragments without staining. A stain plays an important role to see the DNA fragment, that is ethidium bromide, ETBR. So C is the wrong statement. The separated DNA fragments, when stained with ethidium bromide, can be seen in visible light. Visible light is not efficiently useful to visualize the DNA fragments. We need UV transilluminator. UV transilluminator, right? UV light, right? So, and the DNA strands, they can visualize in the form of bright orange color bands. So, here the correct statements were in this A and B only. Fourth option is the correct answer, A and B. Next question. Biological classification question. The main criteria used for 5-kingdom classification proposed by R.H. Whittaker include. So R.H. Whittaker, it is the most successful system of classification. So he classified the living organisms into 5 kingdoms: kingdom Monera, Protista, fungi, plantae, and animalia. The major criteria followed by this classification is cell structure, yes. Body organization, yes. Presence of flagellum, no. Modes of reproduction, yes. Evolutionary patterns, yes. So the answer for this question is A, B, D, and E only. First option is the correct answer. Next one. This question is related to sexual reproduction in flowering plants. They are asking about the ploidy. Ploidy. So central cell, they are asking about diploid cell. Central cell, which is diploid. Primary endosperm cell, it is diploid. Zygote, diploid. Synergid, haploid. So very simple question. It is related to the mature embryo sac, female gametophyte of the angiosperms. So the answer for this question is primary endosperm cell, which is formed after the fusion of vegetative cell with the one of the male gamete. Next one. Next question. Which of the following statements are correct with reference to packaging of DNA helix? This is related to molecular basis of inheritance. So statement A, histones are organized to form a unit of 8 molecules called as histone octamer. Yes. So histone octamer, this octamer contains the below proteins H2A, H2B, H3, and H4, each with 2. So totally 8 histone proteins, they form an octamer, correct statement. Histones are negative charge basic proteins. Histones are basic proteins only, but the charge is positive. Wrong statement, this one. Histones are rich in the basic amino acids. Yes, lysine and arginine, correct statement. The positively charged DNA, here only we can identify whether the statement is true or false, because DNA always negatively charged, wrapped around the positively charged histone octamer to form a nucleosome. So it is also a wrong statement. So the correct statements in this question were A and C. So A, C, E. Yeah, one more statement. Sorry, one more statement is there. The packaging of chromatin at higher level requires additional set of proteins called NHCs. We can also call shortcut NHC, non-histone chromosomal proteins. It's a correct statement. So answer is A, C, and E only. So question number 123, second option is the right answer. Next one, 125. It is also related to molecular based on inheritance. So the question is related to lack of peron. So if you see the lack of peron. So they are asking the question related to these structural genes. Okay, promoter, inhibitor, promoter, operator. So we can call this one as structural genes. So the structural gene Z codes for an enzyme called as beta galactosidase enzyme. Simple answer. Z gene codes for beta galactosidase. Y gene codes for permease. A gene codes for trans acetylase. So the answer for 125 is fourth option is the right answer. Next question. So match the following question related to plant growth and development 2,4-D, 2,4-dichlorophenolactic acid. It is a synthetic auxin which is mainly used as a herbicide, A3. Then GA3, gibberellic acid. It is mainly useful for speed up malting process in brewing industry during the production of alcoholic beverages. So B1. Next one, kinetin. So kinetin, it is mainly helpful for stimulating the stomatal closure. So kinetin, nutrient mobilization. 4. The next one, abscisic acid, it's a stimulator of stomatal closure. So abscisic acid, it can be considered as a natural antitranspirant. So which is mainly helpful for the closure of stomata. So kinetin, nutrient mobilization, it mainly delays senescence. So cytokinins, they are a group of cytokinins. So answer is A3, B1, B1, C4, D. Third option. Third option is the correct answer. So next question. Arrange the following steps of somatic hybridization in the correct sequence. So this question is related to biotechnology applications, tissue culture talk, tissue culture. So let us see here. So isolation of single cells from two different varieties of plants. First one, that one. Next one, digestion of their cell walls. 2. Then isolation of naked protoplast. 3. Then fusion of protoplast to get hybrid protoplast. 4. Then fifth one, growing of hybrid protoplast to form a new plant. So the answer is D, A, B, C, E. Second option is the correct answer. D, A, B, C, E. So the first step is isolation of single cells. Second one is digestion of cell wall by using some enzymes, cellulases. Third one is isolation of naked protoplast. Fourth one is fusion. So the protoplastic fusion, it is mainly carried out by a chemical called as PEG, polyethylene glycol. So chemically mediated fusion, it is mainly carried out by a chemical called as PEG, polyethylene glycol. So last one is growing of hybrid protoplast to form a new plant. So the answer is second option. Next. So C51H98O6 plus 155 molecules of oxygen, 1 or 2 carbon dioxide plus 98 water molecules and energy can be liberated. So this question is mainly related to respiratory quotient RQ. So the RQ of fatty acids always it exact RQ is 0.7. It's about palmitic acid. Next question. Match the following. Yes, biomolecules. Trypsin A enzyme. Morphine B4 alkaloid. Pancanavalin A, it's a lectin. C2, collagen sense. 3, 4, 2, 1 is the correct answer. That is option A. Option A is the right answer for 130 question. Next one, the question related to binomial nomenclature, first chapter. Which one of the following is not true about the universal rules of binomial nomenclature? Both the words and a biological name handwritten, separate, underline. It's a correct statement. The specific epithet in the biological name starts with a small letter. Yes, correct. The first word in the biological name represents the specific epithet. The first word in the biological name, it is generic name or genus. While the second word denotes specific epithet or species. It's a wrong statement. Then biological names, they generally derived from Latin language because it is a dead language. So the answer for 131 is third option is the right answer. Option number 3. Next one. One more question from photosynthesis. So this time in plant physiology, they gave more weightage, they gave more priority for photosynthesis. So one more question that is related to C3 cycle. The enzyme required for carboxylation in the Calvin cycle, that is Rubisco enzyme. Ribulose 1,5-bisphosphate and carboxylase enzyme, Rubisco enzyme. And this enzyme requires magnesium ions as metal ions. Right? So pep carboxylase in C4 plants. Carboxypeptidase. So it's a what we can say it is mainly helpful for the breakdown of carboxylases. Hexokinase, it's a major enzyme in glycolytic pathway, first enzyme. So the answer for 132 is second option, Rubisco enzyme or REBP, carboxylase oxidase. Next one, which of the following formula is the correct floral formula of Solanaceae family? So Solanaceae family is also called as tomato family, potato family, datura and chili family. So in this Solanaceae family, mainly we can observe gamosepalic. Sepals, they are in fused condition. Gamocephalous condition, mainly we can observe in Solanaceae family. So the symbol for fusion bracket. So K5, 5 sepals, they are under fusion. So flower is actinomorphic, bisexual, gamocephalic. Corolla, 5 petals, those petals are fused. Androecium, stamens 5. So we can observe the condition called as epipetalous stamens. When stamens fused with petals, when stamens fused with petals, it is called as epipetally. Gynoecium, 2 carpels, and the ovules they born in axile placentation. So the answer for this question is K bracket 5, C bracket 5, epipetally A5 and G. Here, if you see here, this addition is not there. That's why we need to opt option 1 as the right answer. Here, addition is there, but parenthesis is not there. Fusion is not there. Next one, 134. Which of the following types of pollination brings genetically different types of pollen grains to the stigma? So it is mainly related to cross pollination. So cross pollination in another way we can also call it as xenogamy. So xenogamy, so autogamy, geitonogamy. Third one is xenogamy. It is purely it can be considered as cross pollination. So answer for 134 is third option is the right answer. Option number 3. Next one more match the following. This match the following is mainly related to the sites where the metabolic processes will occur. Glycolysis takes place in cytoplasm, cytosol, A3. ETS, inner mitochondrial membrane, B1. Accumulation of protons, intermembrane space, C4. Krebs cycle, mitochondrial matrix, A3, B1, C4, D2. Second option is the right answer. The next one, 136. It's related to the selection of recombinants and elimination of non-recombinants by technology principles. So insertion of a foreign DNA at bomb expulse site in an E. coli cloning vector, PBR 322 results in the loss of antibiotic resistance towards tetracycline. It is a recombinant molecule. Generally, that vector can lose resistance towards the tetracycline gene. So the answer for 136 is third option is the right answer. It loses tetracycline. Next one, 148. Non-membrane bound cell organelles found in both prokaryotes. Very, very easy. Prokaryotes and eukaryotes. That's why they're also called as universal cell organelles. Next, question number 151. This question is related to bioactive molecules. Streptokinase. The main function of the streptokinase is removal of clots from the blood vessels. A2. Statins. They're mainly useful to lower the blood cholesterol. Next one is lipases. They are mainly useful in detergent formulations. So these lipases, they are mainly extracted from Bacillus and Pseudomonas bacteria. Lipases extracted from two different strains of bacteria. Number one, Bacillus, another one is Pseudomonas. So cyclosporin A extracted from Trichoderma polysporum, it's a fungus. So D1, it is an immunosuppressive agent. So answer is A2, B3, C4, D1. First option is the right answer. This is also one of the simple question from microbes in human welfare. Next question. So this is a question related to cell the unit of life. Choose the correct statements regarding the cell organelles and their inclusions. The endomembrane system includes Golgi, ER. Wrong statement because mitochondria cannot be considered as a part of endomembrane system. A statement is wrong. Rough endoplasmic reticulum bears ribosomes on its surface. Correct. Both mitochondria and plastids have circular DNA. Correct. The DNA found in
Sir,
mitochondria.
Satish sir, the phone has been rotated, sir. Just you can need to adjust the phone, sir.
Okay, okay, sir. Okay, sir.
It's up and down, sir.
Oh.
Yeah, now it's correct, sir.
Okay, sir. Thank you.
Sir, you had muted yourself, sir.
So it's an elaborated proteinaceous network. So D is correct. Mitochondria is a single membrane bond structure. Wrong statement. It's a double membrane bond cell organ. So the correct option for this question is B, C, and D only. Fourth option is the right answer. Next one, 156 question. So this question is related to biotechnology applications. So the toxin proteins isolated from BT, Bacillus, right? Which control cotton bollworm and corn borer. So cotton bollworm and corn borer, Criban right? Cotton bollworm, Criban corn borer, Criban. So answer is third option. Third option is the right answer. 156. So next one. Select the correct statement regarding cell membrane in eukaryotic cell. Yes. A statement, membrane of human RBCs has approximately 52% protein. Yes, correct answer. 40% lipids. Major phospholipids are arranged in a bilayer. Correct statement. Extensions of the plasma membrane in the cell found mesosomes. No, mesosomes they are found in prokaryotes, not in the eukaryotes. Wrong statement. Tends towards the inner part, which is a hydrophobic tail, always inner, and thus protected from aqueous medium. Correct statement. Glycocalyx is present on the outer surface. No, there is no glycocalyx in eukaryotic cell. It is mainly found in the prokaryotic cell, and it is mainly present on the outer surface of the plasma membrane of prokaryotes, not eukaryotes. So it is wrong statement. So the answer for 163, A, B, and D only. Fourth option is the right answer. Fourth option is the right answer. Next question. So this question is related to the classification of enzyme classes. Enzymes, they are mainly classified into six: oxidation reductases, transferases, hydrolases, and this is lyases, fourth class of enzyme. Class 4, lyases, right? So class 5, isomerases, class 6, ligases. So the main purpose of these enzymes are, so without using water molecule, these enzymes breaks down these bonds and leaving double bonds here. Right. That is the main purpose. So the main purpose of these enzymes is they are mainly useful for the production of double bond or ring-like structures without using water molecule. So the answer for this enzyme is lyases, second option. Next one, 178 question. What is the reason behind production of large boars in Swiss cheese? So Swiss cheese, it's a kind of cheese which is mainly ripened by Propionibacterium shermani. So the production of large holes in Swiss cheese due to the release of huge amount of carbon dioxide by a bacterium called as Propionibacterium shermani. So this all about the botany questions from code number 12. Dear students or anyone those who are in this
So
session,
botany
if you are
first.
having any queries, you can ask me.
Only botany.
Okay, sir. Done.
Nice, sir. Nice, sir.
Shall, shall I wind up?
Yes, sir. Yes, sir. Yes, sir.
Oh, thank you. Thank you, everyone, for giving a wonderful opportunity. Thank you, my excellent share college management, for giving this opportunity. Thank you, very much.
Thank you, sir. Thank you, sir.
Uh, Sir, should I stop the live or should I continue, sir?
అనిల్ సార్ అనిల్ సార్
సార్
ఈ కెమెరా రివర్స్ కనబడుతుందా సార్? ఎక్సలెంట్ చేయడానికి క్లియర్ గానే ఉంది సార్ అది.
క్లియర్ గా ఉంది సార్, క్లియర్ గా ఉంది.
ఓకే ఓకే సార్, నాకు ఇక్కడ రివర్స్ కనబడుతోంది.
మీకు రివర్స్ ఉంటుంది కదా సార్, మనకు మిగతా వాళ్ళకి ఓకే సార్.
ఓకే.
కొంచెం సెట్ చేయండి సార్, కొంచెం దగ్గర
ఓకే
లాక్కోండి.
సార్. వినబడుతుంది సార్ ఇప్పుడు? లేదు సార్.
కొంచెం సెట్ చేసుకోండి సార్.
గుడ్ ఆఫ్టర్నూన్ సార్. అనిల్ సార్ చాట్ చేయాలా?
ఎస్ సార్, ఎస్ సార్.
Good afternoon, everybody. Yesterday, Neet 2026 was conducted by NTA. Zoology, no doubt zoology, it is a lengthy paper. But it is not a hard paper. It is a lengthy paper, but not a hard paper. By using some tricks within 25 to 30 minutes, within 25 to 30 minutes, we can complete the paper, zoology paper. It is not a tough paper. Total that paper includes 45 zoology, 45 botany papers. Out of 45, 15 questions are a statement type and 10 questions are matching type. Only 20 questions are MCQ type. But overall, if they got matching type, statement type, MCQ type, 35 are very easy. 35 are very easy. 10 are moderate, no difficult question that we can solve. No difficult question that we can solve. Directly entering into the paper. 96 is a very outline question. Productivity, net primary productivity, gross primary productivity, secondary. Whenever this type of questions comes, first, whichever way very familiar to you. Productivity, rate of biomass production is called what? Productivity. That is what A3. A3, the immediately start of these two. A3, this is wrong and this is also wrong. And net primary productivity, that is a gross primary productivity minus respiration loss. A3B1, here B, A3B1 is also there. Gross primary for the rate of production of organic matter during photosynthesis, that is C4. Here 2 is there, is wrong. Without going to fourth option, we can go. In this way, we can save the time. We can save the time. Then coming to the next question. Since argument diversification of bacteria has been in 5 episodes, 6th episode is now. The previous 5 episodes are pre-anthropogenic, before the origin of human beings, evolution of man. That's why the 6th one now it is which is going on, that is 100 to 1000 times faster than compared to the prehistoric 5 episodes. So how the 6th extinction is different with the previous 5? It is very far. Answer here is the 3, we can directly do. We can directly do. Then coming to next question. The Amazon rainforest forest, this is a question related to biodiversity. What are the consequences of loss of biodiversity? Loss of biodiversity. Here, which is very clear to us. First, we have to select that stellar silicon passenger region has become extincted, because of over exploitation. What is asked in the question? Are correct. This will be there. Immediately we can strike off this one. We can strike off this one. In this way, we can take save the time. And A, B, C. Then Amazon forest being cut down, cleared for cultivation of soya beans, that also correct. Water and then invasive species, that also correct. Nine, here you see, that is not for the growth of the cichlid fish that led to the extinction of cichlid fish. That's why this sea should not be there. Sea should not be there. Then immediately here see that this is the wrong, this is the wrong, and A, B, and E, A, B, and E between the two of these. Then go for when species become extinct, the plant and animal species associated with are not affected. This is wrong. That is what co-extinction. When one of the partner becomes extincted, automatically its partner becomes extincted. That's why answer is what? Second option. Like in this way, we can save the time. We can save the time. Next is the magic type, decomposition, detritus, mineralization, humidification. All are very clear definitions known to us. What is the detritus? Dead remains of plants and animals, including fecal matter. B is the flow. B is the flow. Only one option is there. Here we can say the time. Here we can say the time. Only one second. Here we can have detritus. That is breaking down. B is the 3. Here B is the 3. Here 4 is there. Here 2 is there. 2 is there. Here. Option is what? A3, decomposition. What is breakdown of complex organic matter into semi-inorganic. This is clearly known to us. Clearly known to us. The dead remains of plants and animals, including fecal matter. Mineralization, release of irrigant nutrients by activity of microbes. Humidification, dark color. What is human? That is known to us. Then option is what? See, easily we can go for this. Here it is a lengthy paper, no doubt, but by selecting the question, here direct with which of the following in situ conservation method, here sacred grows, protection of the plants which are of religious importance, that is called as sacred grows. Very direct question, here no need of discussion, this question. Okay. Going to next one. Which one of the following disorders is caused by the substitution of glutamic acid by valine and the sixth position of the beta globin? Two questions came on sickle cell anemia. Here, direct answer is sickle cell anemia. Thalassemia, precursor alpha thalassemia is there, beta thalassemia is there, phenylketonuria, hemophilia, X-linked disease. Here, sickle cell anemia only at position 6 of beta chain. Glutamic acid is replaced by valine. Glutamic acid is replaced by valine. That disorder needs to water a disorder called as sickle cell anemia. That is called sickle cell anemia. Next question, part is regarding what endocrinology, hormonal action, cortisol, that is very well known to the anti-inflammatory reactions. A is 2. And aldosterone, we can add that to what? Stimulate the reabsorption of sodium and water from renal tubules. And polysystokinin, it is secreted by the cells of duodenal mucosa that stimulate the pancreas to release the pancreatic enzymes, and also it brings the contraction in the gallbladder causing the release of bile fluids. Here A2, that is well known to us. A2, 2 are there. A2, 2 are there. Here we can strike off this one. Then aldosterone, that is B3. B3, both are there. Polysystokinin, stimulate the secretion of C4. Here only one is there. Here C1 is, this is the wrong. Here, like in this way, we can save the time. Then coming to next question, arrange the following. It is arrangement, renin angiotensin, it also related to water, nitrogenous base, excretive products and their elimination. Arrange the following renin angiotensin mechanism in the correct order. Whenever the blood flows with a low speed, hypokinetic circulation, when your blood pressure decreases, this extraglobal sensor stimulated that releases the renin and operates the renin-angiotensin mechanism. Renin-angiotensin mechanism. Here first fall in glomerular filtrate. Whenever there is a fall in the blood pressure, fall in glomerular filtrate stimulates the release the release of renin. The renin converts angiotensinogen to angiotensin, that is finally converted to. Second comes this. Then angiotensin is powerful in a vasoconstrictor and release the stimulate adrenal gland causes the release of aldosterone. Then aldosterone increases the previous question we discussed. It increases the reabsorption of water and sodium ions in the distant parts of the nephron. Thereby blood becomes again dilute and that is the final. C is to be first, A should be last. This is the final. This is the final one. Okay. This is the final one we can use. Like in this way, we can save the time, which is very familiar to that we have to first. The next question is a very outline question. It looks at the LND. It is regard to respiration. Respiration takes place in 5 steps. First step, that is exchange of gases between the atmosphere and the lungs. That only called as what? Pulmonary ventilation. That only called what? Pulmonary ventilation. This is one. Then after that, exchange and diffusion of oxygen into the blood, release of carbon dioxide, release of exchange of CO2 across the alveolar membrane. After that, that should be transported to the tissue. That was done by the blood, transport by blood. Then, after reaching the tissues, diffusion of oxygen into the tissue and uptake of carbon dioxide, diffusion of CO2 between the blood and tissue. And finally, what cellular respiration that takes place inside the mitochondria of the cell. That is C, D, E, A, D. A, B, E, A, D. Then coming to next question. Next question is related to health and disease, life cycle of Plasmodium. Life cycle of Plasmodium, it is also very easy, very outline question. Whenever the female mosquito bites a human being, the sporozoites are eating into our blood. This is the first one. This comes what first one. Sporozoites. The female mosquito increase the sporozoite is human bite. This is the first one. Then immediately you can strike out this, Then the sporozoites entered into our blood from there reaches to the liver. This is the second one. First multiplies in the liver, then in the RBC. Here, a parasite essentially livers and bosses and releasing into blood. This is the third one. Already first C. First one A, then B. Here also you can strike off this also. Here only no need of going to fourth and fifth steps. The third step only you can say the time. You can say the time. Then coming to next question. Next question, very direct question, specific receptors that are a synapse are there, but binding of neurotransmitter. This is the synapse. This is the presynaptic membrane, the postsynaptic membrane. Neurotransmitters are released by the presynaptic. The receptors are present on postsynaptic membrane. Fourth is the option. Then what is the probability of both blood group where both mother and both and with A blood group and B blood group both are like as IA and IO and IB and IO. When both are heterozygous, genotype ratio is 1:2:1, which research of blood group, both blood group are 1/4. The percentage is 25%. Percentage is 25%. Directly, if both are heterozygous. When both are heterozygous, 1:2:1 in the body we are studying, the zoology also we are studying. Okay. Recently, we will go to SA. Then here, matching type of question ERV estimated reserve value whenever we release the air by force, 1000 to 1100 ml. 1000 to 1100 ml. This is air is released and the expiratory reserve value. A3. A3 means what? Who are there? This you can strike off, this you can strike off. The reserve value after forcible expiration also whatever the air that remains in the lung that is the reserve value that is equal to 1100 to 1200 ml. That is B4. A4, B4 here it is there. Here B by there wrong. You can go for first option. Like in this way, you can say that I did not in the paper again and again. If the student is pre-prepared like this, they can do the zoology paper within 25 to 30 minutes. Both biology not more than 1 hour, not more than. The student has to prepare that like this. Okay. Then the next question, very correct. Which are the following is not an example of convergent evolution. Convergent evolution means what? Analogous organ shows convergent evolution, and homologous organ shows what? Divergent evolution. A divergent, a clipper, four limbs are based that those are modified four limbs that are example for homologous organ. They are exhibiting what? Divergent evolution. Remaining three, they are example for convergent. Option is what? 3. Next coming to is a very outline question is looking lengthy, but a very outline with regard to frog. Already everybody can knows the frogs are sexually dimorphic. Male frogs consist vocal sacs and a copulatory pad or intestine. Fourthly, okay. Here, vocal sacs are present and the copulatory pad. Here, B and D. Only two sexually dimorphic characters that everybody knows. This is wrong. This is wrong. That is common for both. Common for then male frog is differing with the female frog in having vocal sacs that is B and in having copulatory pad in first digit that is B and D fourth option. B and D fourth option. Like in this way you can do. Next question comes very outline question. A research and a understanding type of question. Here, a group of researchers procured some fish-like animal, like fish-like animal, like this. What is this animal? Everybody knows. Here only unpaired fish present. Fish-like animal, endoskeleton is cartilaginous. By the circular sucking mouth. Circular sucking mouth is the characteristic feature of cyclostomes. Side from the name itself, a tetromedon is the example for tetromedon is the example for cyclostomes. Your answer is what? 2. You have no need of spending much more time on that. No need of spending much more time. Then again matching type, which is familiar to us. That we have to choose first. That we have to choose first. Here, about 65 million in Europe, 350, 320, 3 to 320. This is somewhat complicated D and C which and few complicated. Dinos are disappeared from the earth suddenly from the earth about 350 million years ago. This everybody. Sorry, everybody. Which is familiar to us, to select one. About 65 million years ago, dinosaurs disappeared. Jawless about 50 million years ago, invertebrates were formed. And about 350 million years ago, jawless fish probably evolved. So just now, these are cyclostomes, arthropods, about 320. This is somewhat moderate type of question, and which we familiarly know. These two we familiarly know. First, select that 65 million years ago, what happened, and jawless fish when they appeared. These are the answers. Here, part A2 only 2 are there. Then coming to next question. Again, a matching type of question. It is familiar to all. Here with regard to what contraceptive methods, how they works. How they works. Here, familiar one is the diaphragm. Very easy one. That is a barrier made of rubber used by females. That is everybody knows. C1. C1. In 3 options is there, unfortunately. Next, multi-load 375. And certainly it is familiar. The moral contraceptive page. Once in a week page, D2. D2, here 2 are there. Which one? Immediately you can strike out this. You can strike out. A is progesterone. That is hormone releasing iodine. And multi-node 375. Multi-node 375. Between the hormone releasing and carbon releasing iodine, there is always a controversy. But here, unfortunately, both are closely related. But go with this. Here, A is copper releasing iodine, multi-node, node in that name itself is there. That is loaded with some hormones. Loaded with some hormones. Then easily on that base you can do. Load, multi-load. It is loaded with what progesterone and also what estrogen like hormones. Estrogen like. In that way you have to think. Next is here. It is an application understanding type of question. Very easily we can do. It looks like a tough beat, but it's not a tough beat. WB says count of sample is millimeters. How many eosinophils and lymphocytes in the same blood sample approximately? We know eosinophil percentage is 2 to 3%. 2% of 8,000, like 3 have to do. That is what 160. If 3% of 8,000, then it is 240. Only one option is there. Next office are 160 to 240. No need of counting the lymphocytes that are present 5.3%. Here with this we can do. Here answer is 2. Very easy. It looks like a new type of question, application type of question. If you know the 1% of eosinophils, easily we can do. Easily we can. If we know the clue, easily we can. Okay. Then coming again a matching type of question. Matching type of question. It is regard to health and disease, the alcohol and blood abuse. Here nicotine present in the smoke, present in cigarette smoke, that stimulates the adrenal gland and releases the adrenal gland hormone that is hormone. This is well known to us. A2. A2 means immediately we can strike off. Immediately we can strike off the this one and this one. Third and fourth. Morphine, it is a painkiller. The chemistry we will study and also in the biology. Here, morphine, effective second one painkiller, B3. A2, B3. A2, nicotine, immediately A2. Morphine is what? B3. A2, B3. Only 2 options are next. Here also. Heroin. Cocaine is well known to us. It causes a state of well-being. Then heroin is depressant, slows down. Then answer is what? A nicotine, stimulating adrenal gland, morphine is a painkiller, Aeroid represents protein causes state of well-being and increases the energy level. That the third of this. Immediately we have to do like this. Next question, alpha 1 and fixing. Anybody can do this is used for the protein from non-living animals. That is used for the treatment of emphysema, a respiratory disorder. Respiratory disorder. Next question, ostrich fish, this one bony fishes. Bony fishes. Here, devilfish, octopus that belong to mollusca. Can strike of this. Sawfish, prestige, that is a cartilage fish. It is second strike of this. And next, starfish belongs to echinodermata. You can say the remaining one is flying fish, anglerfish, fighting fish. They are the boring fishes. They are the one. All remaining are well known to you. All remaining are well known to you. Okay. Then coming to next question. Here with regard to what general animal kingdom, general statement related to all phylum. Digestive system in flat elements is incomplete. This is that. Bilateral similarity chief connect the calculation of adult echinoderms are very various in between. We will say by seeing these two options. Here, what is the incorrect statement that are asked? Means what? A should not be there, B should be there. Means if we can strike out this. And A should be very controversy. Pseudo-pseudo is projected by asking this correct. This should so incorrect. This should not be there. See, here C is there. You can strike out this also. Let note of cardiac person throughout throughout the life in the transit. This is correct. B that should not be there. You can strike out the remaining one. Here, first you have to this incorrect one. Means which are correct that should not be there, which are incorrect that should be there. That should be there. Then this is a direct formula with regard to what exponential growth or logical growth or anybody is there, no need of dn by d equal rn, a plus n by a is a direct, no need of discussing this. Next coming to select the incorrect with reference to RH grouping. Erythroblastosis is a common condition observed. Fetus with RH negative. This is wrong. This is wrong. Fetus should be always positive. Mother should be negative. Here, what option? Correct option which is incorrect option. Means this should be there. This should be incorrect statement. Means then you can strike out this one and you can strike out this one. We go to RH and is observed on RBC in the majority of human beings. When majority of human beings are RH, that is correct. That is here that should not be there. Which are incorrect. RH and observed on RBC in the majority of human beings. Before the transfusion, the RH group should also be matched. Let us keep it. RH because absorbed when pregnant mother is RH negative and the fetus is RH positive. These two are the quite controversial statements. Quite controversial statements. Here, which are incorrect? A is incorrect. Remaining are correct. Erythroblastosis fetus cannot be avoided from using RH antibodies to the mother immediately after the second child, not after immediately after first delivery. First delivery. A and B are incorrect statements. Which is most incorrect, which is correct, you choose that and you go accordingly. You go accordingly. Next one, very outline way, is also need of discussion. Molluscans, in mollusk feather like gills are present, gill respiration, that is what bronchial, that is what bronchial. Reptiles only pulmonary respiration. Adult animals respire both pulmonary and through cutaneous. Amoeba, unicellular organism respiring by cellular respiration. A2, B1, C4, D3. Very outline. Very outline. Next, coming to next question. Here related to previous question. That is what sixth mutation one of the causing polymer hemoglobin change in sickle cell anemia. In sickle cell anemia, GAA codes for glutamic acid, codes for glutamic acid. If that is replaced by valine, GAA is coded codes of valine that lead to sickle cell anemia. Whenever oxygen levels are low, the RBC becomes sickle shape. RBC becomes sickle shape. Direct question that we can go. Later question 161 related to what? Related to what muscle contraction. Here, what is asked regarding correct statements with regard to a motor neuron carries signal sent by the central nervous system to the sarcolemma muscle. This is correct. This is correct. Means we can type of this and this. The neuronal signal generates an action potential which causes the release of calcium ions into sarcoplasm. That also correct. That also correct. Increase in calcium ions inactivates, not inactivates, activates acted for the breaking of cross bridges. Actin binds to the myosin head to form cross bridges. This is correct. This is correct. Then we can cite that this also. Answer is no. No need of going to what? Shortening of sarcometric phase by pulling a actin filaments on sliding filamentous theory. Sliding actin filaments slides onto myosin. No need of going to see. Next going to what? It is related to what skeletal system, locomotion and movement. Here, what is asked? Here also correct. Here also correct. We should underline like this. The joint between any two adjoining vertebrae is a cartilage joint. That is correct. Why five cartilage is present? A is correct. As correct is asked. A should be there. A should be there. What? Humans. Okay, sir. A is a human skull is monoclonal. It is dicarbon. It is dicarbon. That should not be there. You can write down this. B1, joint between the vertebral cartilage correct. That should be there. B should be there. You can write down this. In human being, number of cervical vertebrae 7. Most of the mammals, one or two exceptions are there. One or two exceptions are there. C should be there. And we can write down this. And this is the answer. Next, all ribs except all 12 pairs are double-headed bicervical, no exception. That is wrong. The occipital bone of skull is articulated with the atlas vertebra, not occipital bone, occipital condyles. Occipital condyles. Then two is the option. You can go. Okay. Here, it is not again and again. It is not a. It is a lengthy paper, but not a difficult paper. If you read the question now, here only we are getting the truth. We are getting the truth. Okay. Next, spermatogonia undergoes series of certain statements to produce sperm. Select the correct from again. Correct. Here what? Correct. Here, first spermatogonia always undergo meiotic cell division. That is wrong. Spermatogonia divides by mitotic division and forms into spermatocytes. Means a should not be there. You can strike out this one. You can strike out this one. Now we need 2 and 4. Primary spermatocyte divide mitotically. Divide to produce secondary spermatocyte. Here, meiosis one takes place. Mitotically wrong. Mitotically wrong. Yeah. Then you can strike out which are correct. B, C, D. B, C, D. This we can strike off this primary is B also wrong. this answer. By reading two statements only, we got the answer. We got the first two statements are wrong. The first two statements are wrong. Secondary is format through state, secondary where the data produce, top-line format correct, format produce format is formatted transform format models for all are correct. All are correct. By reading two options only, we can go for the answer. Then next question, again related to previous question, Justar normal operators essentially formed by the cellular modification of. This is the nephron part operate arterial entering into this. Here smooth muscle size modified and Justar normal side. And this is PCT, GCT joints and GCT cells and the modified movements together forms juxtaglobal apparatus. That is juxtal convoluted tubule and apparent renal. This is apparent renal arteriole. This is the DCT. Is the DCT. Next one, which one of the following is the appropriate example of sexual disease? It is the population interaction direct question. It is half free sign bumblebee. One of the pattern of the remember with what image that already we studied direct with no need of discussion. It is a memory place. Then coming to the next. Here related to again frog, the frog 3 questions asked. Related to frogs anatomy, Here correct answer of the again. Hepatic total system with a special venous connection between the liver and intestine. Hepatic, that is correct. A is correct. This what A should be there. Then we can strike up this. We can strike up second and third. There are 12 pairs are wrong. Only 10 pairs are previous are present in B also wrong. B also wrong. Then we can A, B. These are one also we can strike up. Remaining with fourth option. Lot of time saving here. Hybrid consists cerebellum and the part of fourth rating. Sinus phenoms and jains are right atrium. That is correct. Yeah. After that, if checking once more, it also takes less time, no problem. Later, this is the we can. It is a only the difficult question to remember and in zoology paper. Entire zoology paper. Peter's movement starts and here appears again and again. How many times we read history, we can forget. We can forget. Here the fetus movement starts and here appears on the head, almost lost. Fetus develops limbs and digits. B 12 weeks of pregnancy. Fetus the external genitalia in the beginning only. That is the 8 weeks of pregnancy. Fetus body is covered with fine hair, eyelids separate and eyelashes. That is what after the end of the pregnancy, 24 weeks of pregnancy, 24 weeks of pregnancy. Yes. This one is our total biology. This one is we can see. Answer is not. Next question. Very easy question. In the x0 type of segmentation. X0. This is ultimately grasshoppers. Here, Korean minus one chromosome less. One chromosome less compared to female. Here, 23 are male. 4 are female. Males and females are respectively directed in one way. In which animal do half-lives and divide mitotically. Here, the as male animals, drones are half-life. They are half-life but not sterile. They are half-life and fertile. Any half-lives that does not undergo meiotic division, by undergoing mitotic division only it produces the sponge. That is what male animals, drones. Male animals are what? Drones option. One. It is very easy and time. Then coming to it is a diagram based question. Diagram based question. Which is the over plasma membrane surrounding over is called about white and line membrane. Surrounding that Gola Peninsula, surrounding that Corona radius. The space between the white and line membrane, this space is called a perivitalized space. Perivitalized space. It is a diagram is there. Diagram is there. Here the layers such are on the chemical from outer, this is from outside to inside, from this side to this side. First, very outside comes to what? Corona radiata. Corona radiata. Means what? See. See, what is the innermost layer? White line membrane. So white line in plasma membrane of the ovum, white line membrane. This should be large, this should be far. Here. Only C part, D not, only one is there. They automatically we can move. Automatically we can move. Okay. Then coming to next question. Very soon I will finish. Ecological pyramids is very, very often anybody only two pyramids are inverted. One is the biomass pyramid with regard to one oceans, another is what number pyramid with regard to a forest ecosystem. Here, which is on the different, which is the inverted pyramid here they are asking. Is the inverted pyramid. Inverted. Here, the pyramid of biomass in sea, ocean that is inverted. Energy pyramids are always straight and upright. Graph line pyramid never can upright. Biomass only two pyramids are inverted. One is what biomass pyramid of ocean seas, another is for forest ecosystem. Forest ecosystem. Next question is choose again the correct one also. With regard to gift, what is gift? If in case many female ovaries are not functional, if that fail to produce the ova, but really fallopian tubes, uterus, all are normal only. The ova connected from the donor that are inserted what? Fallopian tube, granite intrafallopian tube. Here ova are released. Here ova are released into ova are released. Here question this is wrong, this is wrong. It is a transfer of ovum collected from a donor into the fallopian tube. If you know the full form of this abbreviation, what is the answer? Gamete intrafallopian transfer. Gamete that are transferred collected from a donor. Collected from a donor. What is the answer? Next, with regard to what disorders of musculoskeletal system, which is very well known. This is very well known for everyone. Muscular dystrophy, myasthenia gravis, arthritis. This is common thing. Inflammations in joints. Arthritis. Let us see with this. B1. B1. Here D3 is there. Here D2 is there. D3 is there. No need of reading other. Only first option we can go. First option we can go. Like in this way here. Yeah. Okay. Then coming to next. Here this is a sequence. So much every paper, every example, every copy, everybody will write. Everybody will write. Drama Pithakas, from the drama Pithakas. The home habilis one. Oh, you may have Leonata, Emus appear, one year discussing the divorce. Flightless bird with four-legged modified parallel. Flightless bird. Tuthi is the bird. Diaphragm is the bird, but and settle, but these are modern flying birds. They dominate between first and the fifth. The afternoon active for penguins, which are aquatic, they are flightless. Four-limbs are body-like structures suitable for swimming. Shruti was a hostage, fast running bird. Fast running birds. Okay. The last question coming here with regard to population interaction. Again, here what is asked? So the correct statement. Incorrect. All correct statements only Very few are incorrect. Very few are incorrect. All correct statements. Here, first in both parasitism and commensalism, only one species benefit, other species harm. Other species harm. In parasitism, parasite gets benefit, host is affected model. But in commensalism, commensal gets benefit, the host is neither benefits nor harm. That's why first statement is wrong. First statement is wrong. Hold. Wrong. Means what? Here you can strike off one, you can strike off three, and only combination repeat is two. Now go for second. Most specific benefit in mutualism. This is correct. This should be there. This should be there. Okay. What correct is on? Okay. Correct statements asked here. Here, first one is incorrect. Correct in both cases, that is correct. Then this may be correct, this may be correct, this may be correct. We can strike up this. Both species benefits in commensalism. This is the one. Yes, no. Dimension gets benefit, both still neither benefited nor harm. C should not be there. C should not be there. In parasites, only one species benefits and another species harm. This is correct. In organism, one species is harm and other is benefited. This is correct. Then D correct, D correct, E correct. Answer is what? Answer is
Three.
okay. It is a lengthy paper, but within half an hour we can do. Within half an hour we can do by adopting some clues. So, sir, if we are pre-planned, if we have with this type of questions, and we
Yes, sir.
can do it. It is not a tough paper. It is not a lengthy paper. It is not
Yes,
a.
sir. Yes, sir. Lengthy
Thank you.
paper. Yes, sir. Thank you, sir. Excellent
Thank
PowerPoint presentation, sir.
What, sir?
Thank you, sir. Excellent
Okay.
paper
Okay.
discussion, sir. With
As a
help of
public
PowerPoint
speaking
presentation.
lecture,
Yes, sir.
yeah,
Yes, sir.
thank
Thank
you.
you so much, sir.
Anil sir, Anil sir,
Sir.
uh, Anil sir, oh sir, one. Lecture, I think physics. Anil sir is there.
Yes, sir. Yes, sir. There.
Technically, you also got it.
Yes, sir.
రాజశేఖర్ స్టార్ట్ చేస్తున్నారా? రాజశేఖర్
సార్ చెప్పండి.
స్టార్ట్ చేస్తున్నారా మీరు?
చేస్తున్నాం సార్.
రవి సార్, మెయిన్ కాస్టింగ్ ఉల్టా ఉన్నది సార్, అప్ అండ్ డౌన్ ఉంది. ఒకసారి చెక్ చేసుకోరా? కెమెరా సరిగ్గా సెట్ చేయరా?
Yes, sir. Yes, sir. Right, sir.
Then now it is okay.
Yes, sir.
Sir, please confirm.
Sir, just to zoom it to the screen only, sir. We are getting the background as well.
I will inform, sir. Sorry.
Yeah, hello students, welcome to Excellencia. I'm Dr. Pawan Kumar Kasu. I'm going to discuss the NEET 2026 chemistry questions here. So if you look at the question paper, the questions were moderate to easy. Some questions involve some calculations and some of them are some reaction based. So there is some time which is being taken. On the whole, this is easy to moderate paper. If you are scoring around 30 to 35 correct, there are chances of you getting a very good rank in NEET examination. So let's see that what are all the questions that are asked in this NEET paper in chemistry. All right. So now looking at the first question, it is from organic chemistry, the conversion where the cumene isopropyl benzene is converted into phenol. That is cumene hydroperoxide method, is achieved by taking oxygen as the reagent. The second one is acetic acid is converting into ethanol. For that, what you have to do is, first of all, you have to esterify. That means you will be converting carboxylic acid into ethyl acetate, and then you will be subsequently reducing the group so that this entire thing will be converted into CS3 CH2OH. So you can see that it is matching with the esterification. Then the next one is the 1 propanol is converting into 2 propanol. You first do the dehydration. All right. When you do the dehydration, you will be getting the alkene, and when this is added, the water in presence of H plus, you'll be getting secondary alcohol. So you can see that C is matching with option 4, and then lastly you have oleum, which will convert this into benzene sulfonic acid, which is subsequently converted into phenol. So you can look at the option which is perfectly matching with that. That is A is equal to 2. All right. B is 3. Then C is 4. All right. So question number 4 happens to be option number 4. Option happens to be the right answer for the question. Moving on to the next question is again a sequence based reaction. Now we have ethane. When ethane is subjected to the monochlorination, you will be getting ethyl chloride. So this is your X. All right. So this is when treated with ammonia, you will be getting C2H5 NH2. And when it is treated with HNO2, NaNO2 and HCl stands for HNO2, you will be getting what? C2H5OH. Okay. So the major product which is formed in this reaction will be option 2. Organic chemistry sequential questions. Now physical chemistry. Now there is an equation which is given, that is a solid BiOOH, which is a solid, is in equilibrium with BiO plus and OH minus. Now if I take this particular substance and let's assume that the concentration of BiO plus produced is X, then OH minus is also X. Now equilibrium constant for this would be the product of these two divided by 1, because this is a pure solid, is equal to 4 into 10 to the power minus 10. So X is equal to 2 into 10 power minus 5. X is nothing but concentration of OH minus. Now if you know this, POH is equal to negative log of 2 into 10 power minus 5, that is 5 minus log 2, which is equal to 4.7. Therefore, the pH value is 14 minus 4.7, approximately 9.3 would be your right answer. All right. So next, moving on to the next question is again what you call from physical chemistry equilibrium based question. So carbon dioxide is passed through the charcoal and you have got 1 liter was being taken. Now from this some X moles will go give you 2x liters of this. Now it is given that the gaseous mixture is 1.4 liter. So you guys can see that when I find out the X value is turning out to be what? 0.4 liter. That means based on this you can see the composition of the gaseous mixture, the CO2 remaining is 1 minus X, that is 0.6, 1 minus 0.4, and CO would be 2 into 0.4, which is equal to 0.8. All right. So answer would be 0.6 and 0.8. 0.8 of CO, all right, and 0.6 of CO2. That should be the perfect match to this question. Option 3. Next, moving on to the next question about the quantum numbers. You can see when you have n is equal to 2 and L value is 1, you know that L is equal to 0 is S, 1 is P, 3 is D. Okay. 1, then 3 is F, sorry. Then you have D orbital for this. All right. So you can see clearly that you have the orbitals with you. Now 2s is the 2p is the correct matching for this. So A is 2p. All right. So 40 would be 4s. Then 5f would be a perfect match. Then 3d. So you have to match it accordingly. All right. So it's a very easy question. Now when I'm treating this benzene with anhydrous AlCl3, all the hydrogen will be replaced by the chlorine. This is electrophilic aromatic substitution. Whereas the same reaction is done in presence of UV light, the addition would take place to give you gemaxine. Benzene hexachloride is also called as what? Gemaxine. So both the compounds are containing what? 6 and 6 chlorine atoms respectively. So right answer would be 6 and 6. Next, if you take this propyl alcohol and treat it with PCl5, you'll be getting this. Now, what will happen here that when you are treating this with alcoholic KOH, dehydrohalogenation will take place. Now, when you are adding HBr in presence of peroxide, you will be getting anti-Marconi product, all right? Because peroxide leads to what? Anti-Marconi product. So, the right answer for this question, X and Z is being asked, right? X and Z, you have got PCl5. You can see that anti-Marconi H3PO3 and CS3CH2Br. Next, if you look at the question from inorganic chemistry, V2O5 is used as a catalyst in the contact process, whereas iron is used in the preparation of ammonia for Haber process of ammonia synthesis. Next, you have, all right, now when you look at this, A is 3 and then B is 1. The only one thing which is matching with that, then C is 4. All right. So this is the answer to the next question. Now, when you look at ClF3, the structure of ClF3. It has 2 lone pairs and 3 fluorine atoms. Now, trigonal with 2 lone pairs on the chlorine atom. It has a T-shaped geometry with 2 lone pairs on chlorine atom. Looks to be the correct answer for this question. You can see it is called T-shape with 2 lone pairs are being present. Now, when you have this question, you can see the reaction is H2 giving rise to 2H plus 2 electrons. So, EMF of this cell is equal to E0 minus, all right, 0.06 by number of electrons of 2 log concentration of H plus whole square by partial pressure of H2. So, the 2 get cancelled, and when you substitute here the value minus 0.06, you have been asked to find out this value 0.06 log of concentration of H plus value, which is given as 0.02 divided by partial pressure of H2 gas is given as 2. So, you have to simplify this to find the EMF of the cell. Now, moving on to the next question. The spin only magnetic moment, you can see 3D2. It has 2 odd electrons. So, n is equal to 2. So, mu value would be under root of n into n plus 1 Bohr magneton. So, the value would be certainly 2.84. All right. So, easy question. Now, coming on to this, the first product is methyl benzene, and when you are treating this with nitration mixture, you will be getting the ortho and para products. And you know that because of very small difference in their boiling point difference is very small. We always use the fractional distillation technique to separate ortho and para. Always remember this. Okay. Now, there is a bulb rated as 150 watt, converting 8% of its energy into light. So, energy, if energy of 1 photon is given, okay, let there be some X number of photons. So, direct question. And out of this, only 8% is converted into what you call the current. And you know how to find out the energy, even the power is given. Watt into second. You can see per second is asked. Watt into second will give you energy. Power into time will give you the energy, which is 8% of the total energy. So, from here you will be getting the value of X, that is number of protons. Now, there is a test tube containing a salt, drops of dilute sulfuric acid. When you say dilute sulfuric acid, that means category A type of anion, that is carbonate ion, bicarbonate ion, sulfide ion, sulfite ion, etc. And you can see that there is a smell of vinegar. So, you know that when dilute acetic acid is added, dilute acetic acid will be formed, sulfuric acid is added. So, answer should be 1, right? Next, you see the primary nitrile means what? Cyanides. You want the cyanide to be converted into what? The primary amine. You need to pick the reducing agent for this purpose. For this purpose, we'll be using lithium aluminum hydride because it's a polar bond. So, A is there, so rule this out. Okay. Then when you reduce, tin also does the same. Okay. Tin and HCl will reduce only NO2. This won't do it, but I think these two will do it. So, A, C and D should be the right answer for this question. Next, carbon has the ability to form the pi bonds. You have seen it in alkenes. ECl3, boron and aluminum is a monomer when E equal to boron and dimer when E equal to aluminum. That right? Because AlCl3 will form a dimer by virtue of the dative bond with the central atom. So when E is equal to, it is a dimer. Okay, so you have to find out the incorrect statement. These two appears to be. Oxygen exhibit only -2 is the wrong answer, because O2 it is 0, O2 -2 it is -1, O2 -1 it is -1/2. In OF2 you can see that it is +2 also. So there are multiple oxidation states that are possible. That incorrect is option 3. Next you have +3 is most common, but still the cerium shows +4 oxidation state, because you know that cerium is 58, xenon, all right, 4f1, 5d1, 6s2. This is the configuration of the cerium. So what is happening here, that when it is losing all the 4 electrons, it is acquiring what? 4f0 electronic configuration. Lasine test, the element organic compounds are converted from covalent to ionic. You have anything organic, when treated with sodium, the compounds that are produced are the ionic compound, right? Whether that is sodium cyanide, sodium sulfide, sodium chloride. So all these ionic are obtained from covalent. So this is the phenomenal chain that will take place during the Lasine test. So number of hydrogen atoms present in urea. You know the formula for urea, right? So you're given 5.4 gram. That means you need to find out the moles of urea, weight by molecular weight. So you got the moles of urea, and you have to find out the hydrogen atom. So you have to check how many moles of hydrogen is there. You can see 1 mole of urea has 4 moles of this thing. So 4 into this much will be the moles of hydrogen. Then you have been asked to find out how many are present. So into 6 into 10 power 23 would give you the right answer, right? So when you simplify this, you'll be getting the right answer. Next, metamers. You know metamerism, right? Usually ethers exhibit when the number of carbon atoms are varying across the middle functional group. You can see 1 plus 3 is becoming 2 plus 2 is usually the metamerism. Next, you have these compounds like phosphorus and ethyl group and arsenic and ethyl. You have to find out the incorrect statement. Okay. You know, nitrogen forms a triple bond with itself. That's a correct statement. Let us check one by one. Catenation property, you know, phosphorus forms P4 and all that, but catenation decreases down the group. So nitrogen and oxygen both does not have any d orbital, hence this is not possible. Next, phenolphthalein is used as indicator for the titration of sodium hydroxide solution against the standard solution of oxalic acid. The color change that is observed when alkaline pH close to the equivalent point during the titration. So what is happening here, that when phenolphthalein is used to NaOH solution, the color will change to pink, and when you are adding that acid, it will change to again colorless. Now we have the list of compound that you have to identify. Simple questions. You have been given ethene, ethine, methane, and ammonia as it is. So you have to just figure out how many sigma and pi bonds are present. So you can see that ammonia has 3 sigma bonds and 1 lone pair. So ammonia is matching with 2. That is D is matching with 2. You have to figure out only one option to tell the remaining, right? Next. A certain temperature of 500 joule of energy is absorbed, and 200 work done by the system. That means what? Expansion of the ideal gas. In expansion of the ideal gas, how would you write? Delta equal to Q minus W. So you can see that Q is value 500, work done is 200. So delta U is 300. The change in internal energy is asked, which is simple. 300 is the right answer. Now methane in presence of steam, right? Methane when treated with steam, you'll be getting CO and H2, that is water gas. It's a basic question from intermediate textbook. Now we have little lengthy, that you can see it gives 2,4-DNP test. That means it would be aldehyde and ketone. It does not reduce failing. That means it is ketone. So when it is ketone, you have to rule this out. These two options are ruled out because they are aldehydes. Now you can see that when it is oxidized vigorously, it gives rise to what? carboxylic acid, which will give effervescence with sodium bicarbonate. You know that it will give effervescence with sodium bicarbonate. So right answer should be option 2, right? Next, you have copper sulfate electrolyzed, W equal to E by 96,500 into I into T is the formula, Faraday's law. Okay, so you can see the mass of deposited is asked, atomic mass of copper by N factor of copper is 2 by 96,500 into current is 1.5 ampere into time is 10 minutes. That means this many seconds. You have to simplify this to get the next answer. Next, a functional group that can do phthalate dye test. You know, phthalate test, right? Phthalate means you have phthalic anhydride plus 2 moles of phenol. You'll be getting phenolphthalein. And this phenolphthalein will be converted into pink form when treated with the base. So, phenolic hydrogens are detected by the Thalaine's test. All right. So, next, the correct statement, it's a simple theoretical question. A double strand helical structure and contain thymine as one of the, thymine is a very important thing to distinguish what you call DNA and RNA. So, direct question. Then molarity based question, you have to do little calculation here. So, molality, this is lengthy, is weight by molecular weight of ethanoic acid by weight of solvent in kilogram, right? So, this is the molality. Then molarity is asked. You can see molarity formula is number of moles of solute, that is 5 by 40 by volume is 0.450 liter. You guys have to check this. Okay. Aquatic species are more comfortable in cold water. That means what? Oxygen solubility is more. That means fish and any sea dependent or aquatic dependent life is more comfortable in cold water because solubility of oxygen is more. Whereas in hot water, oxygen gets evaporated, and you know that these fishes die during the hot summer. So these three looks to be all right. Solubility of gas increases with decrease of pressure is wrong. It is in contrast to Henry's law. It should increase with increase of temperature. Then number of moles of B it should be, but this is wrong. So looks to be that A, B, C are correct answers for this question. Next, mixture of chloroform and acetone form a solution with negative deviation for the reason that they form hydrogen bonding. All right. Next, the buffer solution contains, buffer equation, right? pH equal to pKa plus log concentration of salt by weak acid. So you can see that these concentrations are equal. So you'll get log 1 is equal to 0. So pH would be pKa. So pKa is 10. So right answer is 10 because you are given the value of what? The K. Next. Incorrect statement you have to identify. IUPAC 107. Nil septium. That's correct. Largest and smallest species among the following are given. Largest would be, okay, makes sense, aluminium, because the orbit goes into 3 also. And shortest will be Al plus 3, not Mg plus 2, because more the positive charge, smaller is the size. So incorrect is option 2. The correct order of metallic character, right? You know that sodium is more metallic. Then comes magnesium. Sodium more than magnesium. Then beryllium is a metalloid. Okay. So 35 is a theoretical questions. Next, IUPAC name. When you are naming it, you should number from here. So 3-ethyl, 5-methyl, heptane is the right answer. I hope everybody had attempted it correctly. Now, when you look at these compounds, you know that platinum compounds are square planar. Always remember this. All right, and this has a ligancy of 6, so definitely octahedral. Then this is tetrahedral because there is a weak field ligand, sp3 hybridization. Then remaining is trigonal bipyramidal because the denticity is 5. So you can see that what is matching with that. All right, so A is 3. A is 3, then B is matching with 1. So you can see option 1. Next, 38. The concentration versus time. You know the equation, right? You know K is equal to 1 by T into n minus 1 into 1 by 80 whole power n minus 1 minus 1 by a naught whole power n minus 1. So when I substitute n equal to 0 here, I'll be getting an equation where I'll be getting what you call rate versus concentration is turning out to be like this, something like rate is equal to concentration is equal to, that is, you can write in terms of AT, you'll get a minus K naught into T. So I'll be getting plus A naught. A straight line with negative slope for the zero order reaction. Next, ambidentate. That means more than one donating centers. You know thiocyanate, right? SCN. This could be written as SCN or CNS. Okay, carbon can be the donor, cyanate. In this case, it is thiocyanate because there is sulfur, right? This is isothiocyanate. So there can be two different donor atoms. So right answer is option 4. Next, you have the next question. So delta G, correct option with delta G negative, right? Delta for the reaction, spontaneity of the reaction. So what is delta G? Delta H minus T delta S. Substitute both the values and you need to check. All right, delta H is minus 10, minus of temperature is given as 298, minus of minus is plus into 44. So this value is turning out to be what? Plus value, and hence it is non-spontaneous. Maybe it is turning out to be this value. Next, when the element is having more number of bonds than the valency, it carries positive. Less number of bond than valency, it is negative. So you can see 1 is positive. On 2 it is 0 because its valency is satisfied. So on 2 they asked first, so 0. On 1 plus 1, right? 0 plus 1 and minus 1. Next question. You have to write Kp equal to, Kp is not equal, right? You know, Kp equal to Kc into RT whole power delta n. If delta n is equal to 0, then we say Kp is equal to Kc. No, you have to see in which case delta n is equal to 0. Over here, delta n is number of moles of the gaseous products minus number of moles of gaseous reactants. So this is 0. So Kp equal to Kc here. Here also 2 minus 2, 0. Here also 2 minus 2, 0. That means except this, all the remaining cases Kp is equal to Kc. Next, let's look at the next question. Now you need to compare this with the Arrhenius equation which says this. So you can see that from this, the activation energy, this is the previous JE question also. So EA by R value is given 1.25 into 10 to the power 4. If you compare, this value is EA by RT. So this value is given from here, EA is equal to 1.25 into R value. You have to appropriately submit because you are given the answers in calories. So you need to approximately substitute. Okay. So this is the value of this thing in kilocalories. You have to find out. So you can see it is 2.5 into 10 power 4 or approximately 24.84 you will get. Okay, 25. Now you have the next question. C2H5Cl you want to convert into Z, and that Z, how would you get Z? This is Hartmann bromamide reaction where Y is C2H5NH2. Then cyanide, isocyanide test where you will be getting NC. So you want isocyanide. You know how do you get isocyanide from ethyl chloride? By treating it with what? Silver cyanide, but not potassium cyanide. So you have to take silver cyanide and you will get isocyanide. Next question. You can see that you have been given various this thing. All right. So you can see linkage. That means two different ligands. So C matching with 4. You have to check only one option to get the remaining. You see C matching with 4 is only found with this option 3. That's all. Thank you for watching. Thank you.
Sir, do we have any other, sir?
Okay. So hello all. I'm here to discuss 2026 NEET physics paper, and let me tell my opinion about the paper analysis. What kind of paper I'm finding this. So this 2026 NEET paper is basically you can put in moderate and to some extent lengthy also. Kind of 20 question out of 45 where will come in under easy category. Rest 20 question will come into the moderate, and 5 to 7 questions are approximately a bit lengthy calculations are there. Moreover, there are many places there are unit conversion and all that we need to take care. We've got to be careful about those things. And overall, if you just look at the paper, it covers almost all the area in the physics. Mostly it is capturing from the 12th portion, but it has a good number of questions from 11th also. It is covering mainly the mechanics, thermodynamics, ray optics, wave optics, electrostatics, modern physics. These areas, it is no number of questions are covered. So without any delay, let's go to the questions and discussion on that. So this is the first question. So 100-turn closely wound circular coil, radius 5 centimeter, magnetic field is given, and we need to get the current and the magnetic moment. So this kind of things are there, like this is the coil kind of thing. There are many turns, let's say 100 turns are there, and with the radius is given to be 5 centimeter. So we know that B is equal to how much? If n turns are there, n mu naught I upon 2R. This is the formula. They are asking for the current. So they are asking the current flowing through the coil and the magnetic moment. These two we need to answer. So we know the relation magnetic field at the center is equal to n mu naught I by 2R. So B is given. So I can be calculated from here. It will become B into 2R divided by n into mu naught. Right? If we put all these values are given here in SI unit. So B is 3.14 into 10 power minus 3. 2 radius is 5, convert into meter. Divided by number of turns are 100. And mu naught is 4 pi into 10 power minus 7. If you solve this, you are going to get 5 will get cancelled, and 10 power minus 5. After solving this, you are going to get 2.5 into. Okay. So that is the answer for this question. Here 10 divided by 4 will become 2.5 ampere. Now, if you want to calculate the magnetic moment, we know that this is just formula based. It's equal to NIA. Number of turns are 100. Area is 2.5. Current is 2.5. We are calculating from here. And area is going to be 5 times R square, which is 5 into 10 power minus 2 whole square. So if you solve this, it will come out 1.96 ampere meter square in SI unit. Okay. So the answer is going to be first option. Let's do for the next question.
Here we need to do the matrix matching kind of questions are there. So first is E is equal to h nu. This is the photon energy. Okay. So energy of the photon. So first we go to the fourth. Diffraction and interference. These are the wave properties. So wave nature of light. Lambda is equal to h by p is the de Broglie wavelength. We know that. And Crompton effect is particle nature of the light. So these are going to be the. So one we can say that A4 B3. So A4 B3 is here also. A4 B3 is here also. Then C is 1. So C is 1. Option 1 is correct in that case. Okay. Here, the current I in the circuit shown below, our diodes are ideal and identical. So we know that if an ideal diode is there, in forward bias, its resistance is 0, and in reverse bias, it offers infinite resistance. So we are going to observe which are the reverse biased and which are forward biased, which are the branches, because all these branches are in parallel. So you can say this is going to be the reverse bias. So this will become an open circuit. And here also it is a reverse bias. It is going to be the open circuit. Only these two branches, this and this, is going to be the forward. So this simplified circuit will become 4 and 2 is going to be parallel. So R equivalent is going to be R1 R2 upon R1 plus R2. Four by 3, right? And then the current. See the equivalent resistance of this circuit. The current is going to be total potential which is going to be the 30 by 4 MP. 30 by 4 can be also written as 15 by 2 MP. So this is the answer. We can go for the next. The speed of the light in vacuum is taken as unity. You are taking as a unity the speed of the light. If light takes 6 minutes 40 seconds to reach the Earth from the sun, the distance between sun and the Earth is in the new unit we need to know. So we know that distance is equal to speed times time, speed into time. Okay. Speed is equal to 1 unit in the new unit, and into time is going to be in 6 minutes 40 seconds. The total time will become 6 times 60 plus 40, which will be 400. So in new unit, the distance is going to be 400 units, right? The following plots show the variation of the velocity with time of the ball thrown vertically upward and falling back. Which of the following plots are correct? Okay. So look, here is a ball that you are throwing it upward. It will go, velocity will keep on decreasing, will become 0 at the highest point, and then it will change the direction. So if you are taking, generally by default we take upward positive. If you are taking this as a positive, the other half will become negative velocity. And at the highest point, the velocity will become 0. So if you look at the graph will be, the most suitable graph will be starting from positive. It will be going a straight line downward, it will become 0, and then it will become negative. Moreover, the velocity is given by initial velocity u minus gt. So you can compare from here, the slope is negative, and this velocity is initial velocity u, and the slope is equal to minus g. Okay, so this is the most appropriate. So answer is C1. This is about the instruments. The vernier calipers 20 VST coincide with the 16 NST. So we know that, let's calculate. We know that they are asking finally the least count of the vernier calipers. What is the least count of the vernier calipers or any instrument? So the least count of vernier calipers is equal to 1 MST minus 1 VST. The value of 1 division on main scale minus the value of 1 division on vernier scale. So let's first calculate what is the value of the 1 division on vernier scale. So we know that 20 VST is equal to 16 MST. So 1 VST can be written as 16 by 20 MST. But 1 MST is given each division of length how much? 1 millimeter. So this can be also written as 4 by 5 and 1 MST is equal to 1 millimeter. So that is equal to 0.8 millimeter. So the value of 1 division on vernier scale is 0.8 millimeter. And hence the least count will be 1 MST minus of the value of 1 VST, which is going to be 1 MST is 1 millimeter minus 0.8 millimeter, 0.2 millimeter. The same 0.2 millimeter can be written as 0.02 centimeter. So answer is 3. An AC circuit contains resistance 1 kilo ohm, capacitance is given, inductance is given. The resonance frequency we need to calculate. This is again the formula based. We know that resonance frequency is equal to 1 upon 2 pi under root LC. Calculate it. L value is given 1 millihenry. Convert it into SI unit, 10⁻³ Henry into C value is 0.1 into 10⁻⁶, which is going to be 10⁻⁻6, right? So it's going to be finally 10 to the power minus 10 is square root 10⁻⁵, 10⁵ divided by 2 pi. Okay. If you solve this, I can write like in kilohertz they are reporting. This is going to be in hertz. So you can convert like this 100 divided by 2 pi kilohertz. And this if you solve, it will become 15.9. Right? The figure given shows a long, straight, solid wire of circular cross section of radius A. So this is the wire with radius A, and this is a solid section complete. And current is completely distributed over all along this, all along on this area, right? So in such cases, we use the Ampere's law to calculate that, and we know that what we get current at center. I mean, at axial point, we get it, sorry, magnetic field at the axial point, we get it 0 at axis. And from axis to boundary of the wire, circumference of the wire, it goes linear variation. It will be keep on increasing. And then once the point is outside of this, it will go 1 upon R. It is proportional to 1 upon R. And this side, B inside, it is proportional to simply R. So this is the straight line, and then it is proportional to 1 by R. So the option 1 is correct. Okay. Now, uniform metallic wire having resistance 4 ohm is bent in form of a square loop ABCD. So this is A, B, C, D. This total resistance is 4 ohm. It means they are of equal length. So each one is going to have the resistance 1 ohm, 1 ohm, 1 ohm, and 1 ohm, right? Now, a resistance of 2 ohm is connected between points B and D as a diagonal kind of, and a battery of 2 volt is connected here between A and C. Now the value of current is, we need to find the total current, right? So if you see, observe it carefully, this is exactly similar to the balance with stone base. if you just little bit change it, A point will come this side, so it can easily be redrawn like this: A to B, B to C, and then here this can be changed like this. Here it is 2 ohm. This is point is A, and this point is C. This is B, and this is D. All these are 1 ohm, right? And the battery is connected here, which is 2 ohm. So it is the balanced Wheatstone base. We can ignore this and calculate the equivalent total. This 1 and 1 you can take as a series, so 2 ohm. And here also 2 ohm. 2 ohm and 2 ohm in parallel will become 1 ohm. So total current is equal to I is equal to total voltage 2 volt divided by 1. So the answer is 2 amp. Okay. Unknown nucleus has a nuclear density this much, and mass of nucleus is given this. Its mass number A is approximately how much? Okay. So we know that the total mass is equal to density into volume. So that can be written as density of the nucleus into volume is going to be 4 by 3 by the radius. If R is the radius of the nucleus, if this is the nucleus and it has a radius R, but we also know that this radius of the nucleus is R naught, A to the power 1 by 3. If we put it here, I can write M is equal to density times 4 by 3 pi R naught whole cube times A. So A, we need to calculate A. This is A is the mass number. A is equal to total mass of the nucleus divided by density times 4 by 3 pi R naught cube. You can put all the values, like M is given to be 19.926 into 10 power minus 27 kilogram. Density is 2.29 into 10 power 17. All are given in SI unit, so no need to change any unit. Into 4 by 3 pi, and R naught value is given to be 1.2 into 10 to the power minus 15 whole cube. If you simplify this and calculate it, it will come equal to 12. So the correct answer for this is 12. Let's go for the next question. A rectangular wire loop upside 8 centimeter and 3 centimeter. So this is the loop, let's say, with a small cut is moving out. Let's say here is a small cut, and it is moving out. Let's say magnetic field is perpendicular like this. And it is taken out with velocity v given as the b value from where it is coming out is 0.3 Tesla. So it's moving out of region uniform magnitude 0.3 Tesla directed normal to the plane. This is the plane and it is directed normal. The EMF developed across the cut. We need to calculate the EMF developed across this cut. If the velocity is given to be 2 centimeter per second. This is 2 centimeter per second. Okay. And it is moving perpendicular to the shorter side. These sides are how much? This is given to be 8 centimeter. And this side is given to be 3 centimeter. Right? Now these two sides, we just see these length sides. These length vector is parallel to the velocity. So they are not going to induce any EMF. Only thing left out is this. So EMF induced is going to be BLV. BLV, right? So B is going to be how much? 0.3. But length we need to take this one, breadth only, which is 3 centimeters. So 0.03. And into velocity is 2 into 10 power minus 2. Right? So it will come 18 into 10 to the power minus 5 volt. And this also can be written as 1.8 into 10 to the power minus 4. So option 1 is that. Okay. This is a galvanometer of resistance 100 ohm gives full deflection or a current of 1 milliampere. It is converted into an meter which can measure 10 ampere current maximum. The shunt required is, so we know that if this is the galvanometer, we just connect a shunt, a small parallel resistance to convert into a meter. Now total current is coming from here is 10 ampere maximum. At that case, it should be give the full deflection, means in the galvanometer, the current should go how much? Maximum possible, 10 to the power minus 3 ampere. And this side, how much it should come? 10 minus 10 to the power minus 3 ampere by using the junction rule here. And let's say that this resistance is smaller, that we need to calculate. This resistance is given to be 100 ohm. Since these 2 points, these both galvanometer resistance and the shunt resistance are in parallel, potential difference should be same. So 10 to the power minus 3 into 100 must be equal to, this is approximately 10 ampere only. So 10 into resistance is small. So if I just do that, it will become R is equal to 10 to the power minus 2, which is 0.1. So the answer will be 1. Okay. This is on YDSC. Young's double slit using monochromatic light of wavelength lambda. The intensity of light at a point on the screen where the path difference is lambda is k. So you know that wherever the path difference is integer multiple of the lambda, there we get the constructive interference and intensity will be maximum. So this is the maximum intensity, basically maximum possible intensity on the screen. The intensity of light at a point where the path difference is lambda by 3 will be. So we know that intensity at a general point is equal to the maximum intensity on the screen cos square phi divided by 2. Where phi is the phase difference at that particular point on the screen. So we need to get the phase difference to calculate the new intensity. And here we have from the question I max is equal to k. So I can write I is equal to k times. Now, this is the path difference at that point where we need to calculate the intensity. We know we can calculate that there is a relation between phase difference and path difference. Phase difference is equal to 2 pi by lambda into path difference. Path difference is given lambda by 3. So it's 2 pi by 3. This is the phase difference. We can put it here to get the intensity. It will become, if we divide by 2, 2 will get cancelled and it will become pi by 3. Cos 60 is equal to 1 by 2, 1 by 2 whole square. So k times 1 by 2 whole square, which is equal. So intensity at that point will become k times 4. Okay. The magnitude and direction of acceleration produced in a body of mass 5 kg when two mutually perpendicular forces 8 Newton and 6 Newton act on it. Let's say this is the body of mass 5 kg and these are the two forces acting on it, 8 Newton and 6 Newton, perpendicular to each other. Of course, the resultant is going to, the net force is going to act like this, parallelogram rule. This is going to be the net force. Net force is going to be equal to root over 8 square plus 6 square, which is 10 Newton. So how much the acceleration mass is given to be how much? 5 kg. So acceleration is going to be equal to net force divided by the mass, which is 10 by 2 meter per second square. Now let's calculate the direction theta from the 8 Newton. So we can say that theta is equal to tan inverse perpendicular 6 by 8, which is tan inverse 3 by 4. So it's making an angle tan inverse 3 by 4 from which force? 8 theta. From 8 theta. Let us check. And acceleration is 2. Tan inverse 3 by 4. Tan inverse 3 by 4 with 8 Newton and acceleration is option K square. Okay. Here are the 5 capacitors. All these C1, C2, C3, C4, they belongs to one single branch. Means they are in series. And between these 2 points A and B, the equivalent of all these is in parallel with C5. Right? So let's first calculate the equivalent of all these C1, C2, C3, C4. So it's going to be 1 by C is equal to 4 times. It's going to be C will become 4 by 10 due 10 by 4, which is 2.5 microfarad. Okay. So you can remove all these, and in place of that, we can put one capacitor and one will be there as it is. This is 2.5. C5 is also given how much? You can see 2.5. So here also the equivalent 2.5 and here also. And now this is complete thing is connected with 50 volt pair. Right? The equivalent of this complete is going to be 5 microfarad. We can add it. So C equivalent is going to be 2.5, which is 5 microfarad. And hence Q can be calculated is equal to C into V, which will become 5 into 50, 250 microcoulomb. So total charge is coming to 50 microcoulomb. You can directly calculate the charges on each one because both are in parallel. So potential difference will be across this also 50 volt, across that also 50 volts. So it will come 125 microcoulomb here and 125 microcoulomb. So on all the capacitor one way, since on this all series equivalent is getting 125, in series all the capacitor will get the equal charges. So all C1, C2, C3, C4 are going to get the charge 125 microcoulomb, and this rest C5 also is going to get the same 125 microcoulomb, right? So we can say they are asking equivalent capacitance and the charges on each capacitor. So equivalent capacitance is how much? 5, and charge on each one, all these capacitors are 125. So we can have the answer like here looks like there is some slip in that, on all the capacitors. On all capacitors. So this option should be. So answer is C equivalent 5 microfarad and 125 microcoulomb on each. In a meter bridge experiment, the position of cell and galvanometer are interchanged. We shall observe in this galvanometer. Like if it is balanced one and then you are changing. So we know that if the Wheatstone bridge is balanced, meter bridge works on the principle of the Wheatstone balance, Wheatstone bridge only. So if it is balanced one and then you are interchanging galvanometer and cell, there is not going to be any effect on the deflection. It is going to remain as it is. However, it is not mentioned here. It is not mentioned that it is balanced or not balanced. So we cannot consider that as a balanced only. Now, if so, and you don't have the idea about R1 and R2 also. So if you are going to interchange, it can, if it was unbalanced and you are interchanging, it may go any side, depending on the R1 and R2. And so it may go towards left side also, right side also. But in case eventually, if it is at balanced condition and then you are interchanging, then it's not going to have show any change. So what should be our answer? It can happen both right side and left side deflection, and at balance point, no deflection. So this is the most appropriate answer out of the given. The power of a crane which lifts a mass 1000 kg to a height of 20 meter in 10 seconds. Basically, they are looking for the average power. Average power is equal to, we know that, work done divided by time, which is how much work needed to be done. mg, you are working again against the gravity. If you are taking it up, mgh is the work done divided by time, and you can calculate it. So this 100 and 0 will become 1000, and this 98 point, this will become 19.6. 1000 can be written in kilowatt. So answer should be 19.6 kilo. Okay. Again, there is one question on the matrix matching. Young's modulus. Young's modulus is related to the stress by strain. We know that. Then compressibility. In compressibility, it is the inverse of the bulk modulus. So it is going to be this formula. This is the inverse of the bulk modulus. Bulk modulus formula is going to be the, here it should be changing the pressure actually, but it's fine. It's written as excess pressure only P symbol. So that is the bulk modulus, and Poisson's ratio is that is the lateral strain divided by the longitudinal strain. So that is the, so these are the options that we can just take that A2, what we are getting A2, then we are getting B3, then we are getting C4 and D1. So option 4 is the same. Okay. In a concave lens, a ray of light emanating from the object parallel to the principal axis of the lens after refraction. So look, this is the concave lens. This is the principal axis, and from object it's coming, rays are coming parallel to the principal axis, right? We know that once it falls, this is the diverging lens. It gets diverged, and if you just reverse it, it will be coming from this focal point is called principal focal or second focal. This is known as F2. Okay. So what are the options we can take? Emerges parallel to the principal axis? No, it is not emerging out parallel. Appears to diverge from the first principal focus. Ideally, it should be the second principal focus, not the first one, by the naming. Okay, conceptually. Passes through 2F, which is the radius of curvature of the lens. No, passes through the second principal focal. So the most appropriate answer is actually this is diverging, right? This is diverging. So only thing, there is a little chance of getting bonus also here, but the most appropriate answer is 2. Okay, because appears to diverge from this should be the second principal focus, ideally. Okay, conceptually, but they have given first principal focus. But out of the given option, this is the most suitable one. A thin wire of length L and linear mass density small and bent into the circular ring in XY plane with center C. The moment of inertia of the ring about an axis y dash. We need to, it is a kind of a tangent, right? It's tangent. About this axis we need to calculate. So to reach there, we need to first calculate about this axis. We can calculate about this axis what we usually recall about the ring. If it is a ring of mass m and radius r, we know that if this is the axis perpendicular to the plane, the I is given as mr square, right? Now, if you use the perpendicular axis theorem, since these two are equal, so this axis, moment of inertia about this axis will become, let me say it I1, it will become mr square by 2 by using the principal axis, the perpendicular axis theorem. Once you reach there, you can use this distance is r, you can use the parallel axis theorem. So finally, let's say I y by dash. This I1 can be written as, this is I1, it's I center of mass, it's passing through the center of mass. So it is equal to, we got mr square by 2, this is the I center of mass, and plus the parallel distance is equal to mr square. Finally, it becomes 3 by 2 mr square. Now, they have not given direct input as a total mass of the ring and radius of the ring. Rather, they have given the mass density, mass per unit length, and the length. So, total mass is going to be mass per unit length into the length, and the same L length is bent into the circular radius of R. So, circumference must be equal to length. So, length is equal to 2 pi R. That R can be written as L upon. We can put all these things. So, moment of inertia will become V by 2 ml into, finally it will convert into 3 by 8 ml cube divided by 5. 3 by 8 ml cube divided by 5. Option. Okay. Each side of a metallic cube of mass of the metallic cube is given. Some cube is there, right? The mass of this metallic cube is given this much kg, and its side is given to be 9.0 centimeter. Keeping the significant figures in view, the density of the material of the cube can be best expressed as where value of x is. So we need to take care of the significant figures. We know that this we need to calculate the density, and density is equal to mass divided by volume, right? How many significant figures are there here in this mass? 1, 2, 3, 4. There are 4 significant figures, right? And how many significant figures are there in the length? There are 2, because decimal 0 is given, means there are, this is also significant. So 2. Now, if you are going to divide this, so we know that volume is equal to L cube. So mass is equal to 5.580 divided by volume is equal to L cube, which is 9 whole cube. Okay, 9.01. So we can calculate all right. If you will do it, will solve it, it will come like this 7.654 something into 10 to the power minus 3 kilogram. Now we need to take the best expressed it in proper significant figures. So how to decide what should be the significant figures in this? We need to take, in case of division, we need to take the least significant figures. So least significant figures is how much? 2 here. So we need to round up it to 2. So it will become 5, we'll make it in this one. So it will become 7.7 into 10 power minus 3. So answer should be 7.7. Now, for a traveling harmonic wave, this is the equation given. X and Y are in centimeter and this is in second. The phase difference between oscillatory motion of the 2 points separated by a distance 0.5 meter. Okay. Now, if you want to calculate phase difference, so there is a relation between phase difference and path difference. Phase difference is equal to, we can also write like 2 pi by lambda into delta x, or you can also write k times delta x. Right? From wave equation, we can directly get the k value. Coefficient of x, all these values, if you take everything inside, 2 pi into 0.00, that is the k value. So 2 pi into 0.008. And how much is the delta x value? 0.5. Okay. So what will be the answer approximately? 0.5 into 2 becomes 1. It will become 0.008 by rate. So option 3 will be correct. Okay. Passing through this is equilateral prism. So prism angle is going to be 60 degree. This QR is parallel to BC. It's given. Now, if so, this is the normal is going to make here this angle and this angle is equal to, this is 60 degree. So this is going to be 30 degree. 30 degree. So what I can say, R1 is equal to R, and which is equal to how much? 60 degree. Okay. The angle of incidence is given in the question. It is 50 degree. So we know that when R1 is equal to R2, I is also equal to E. In that case, it's going to be I is equal to E is equal to how much? 50 degree. The angle of refraction of this prism is given. Since it is equilateral, 60 degree. So in that case, delta is equal to, we know that I plus E minus E, which is how much? 40. So the angle of deviation is going to be 40 degree. In the circuit shown below, the voltage appearing across the diode D. Look, here we need to get the voltage, not here across the resistor. So in first half loop, this is going to be the positive and this is going to be the negative. This diode going to be in reverse bias, and we know that in reverse bias, the diode offer infinite resistance. So total potential, whatever you are applying, is going to be you are going to absorb across the diode in that case when the resistance is infinite. So if you take the first loop, along output is also going to be the first loop. In the second half, this will become positive and this will become negative. It will become forward bias. In forward bias, the resistance across this diode is going to be 0. And since resistance is going to be 0, potential difference is also going to be 0. Next half is going to be. So, which one is the option? Option 2 is the correct one. Okay. For a simple pendulum having time period T, the variation of kinetic energy with time. You know, like if you, we know that kinetic energy is equal to mv square one way. And depending on what, you know, like you have taken the equation, x is equal to, let's say, if I am taking a sine omega t. Okay. Or let's say if I am taking x is equal to a cos omega t. You can take either one. Then v will become a omega sine omega t. And in that case, k is equal to m. This is now there. Okay. Half m a square omega square sine square omega. Okay. We are going to graph this. Since it is a square term, it is going to be quit after half time period. Okay. So this is the most appropriate answer in this particular case. Okay. So kinetic energy we are starting 0 means it is we are just thinking that it is starting from the extreme position. Here at T is equal to 0, it is starting from x is equal to a. Then kinetic energy will be 0. At mean position, it will reach maximum. And then again, it will never go negative because kinetic energy is never negative. It is always positive. All right. Our resistor is connected to a battery of 12 volt EMF. So here is the 12 volt battery and internal resistance, and it's connected with some resistance, external resistance. 12 volt, internal resistance 2 ohm, and the current flowing is 0.6 ampere. The terminal voltage of the battery. Terminal voltage is potential difference across these battery. This is called terminal. VA minus VB is called terminal potential difference. And we know that this is the case of discharging of the cell. So in that case, we know that VA minus VB is equal to E minus I times R. $10.84 is going to be. All right. The amount of work done to reach a mass m from the surface of the earth to a height equal to the radius of the earth. Okay, so the situation is like that. If this is the earth, you are taking a mass from here, from the surface of the earth to equal to. So if you are an external agent and you are taking a mass of m from the surface of the earth to a height equal to, this height is given as, from the surface to this height is given as. So they are asking amount of work done by external agent. We know that work done by external agent is equal to final potential energy minus initial potential energy. This is the final configuration, mass and earth. So it's going to be, we know the formula, minus G mass of earth, small m is the mass of the body, and the total distance from the center is 2r, minus of U initial is going to be minus GMM. Initially mass was here at a distance r from the center. So it's going to be it will become, but this GM can be written as small g into r square because we know that G is equal to GM by r square. So GM can be replaced with the g into r square. So the answer is going to be mgr divided by 2. Right? An electric heater supplies heat to system a rate of 100 watt. If the system performs work 75 watt, joule per second is watt only, then the rate at which internal energy increases will be. We know that from the first law of thermodynamics, Q is equal to W plus U or delta U. So if you can just dQ upon dt is equal to dW upon dt plus du upon dt. Okay. So we need to calculate rate at which internal energy increases. Du upon dt. U is the internal energy here. So we can say that, and here what are the input given? Input given is dq upon dt is given 100 watt. This is the heat supply rate. And rate of work done is given 75. So you can calculate du by dt is equal to 600 minus 75, which is 25 watt. The answer will become, okay. A room heater is rated 400 watt 220 volt. If the supplied voltage drops to 200 volt, what will be the power consumed? You know, these ratings gives the idea about the resistance of that coil or bulb or heater anything. So how to calculate that? We know that R is equal to V square by P, right? So the resistance of this heater is going to be 220 into 220 divided by 484 ohm, right? Now, if this resistance, this heater is connected with a new power supply, which is how much? 200 volt. Same heater. Then what is the power loss to this? Power loss to this. The same potential will go across this V square by R. Right? If we solve it, it comes around T31. All right. When a ruler falls vertically, 5 different person catch. You know, in the reaction time experiment, we do the same thing. We release a ruler and somebody catches it. And based on that, we measure the reaction time and compare the reaction time from one person to another person, etc. Okay. So, different persons' reaction times are listed here. They are asking what is the correct order of the distance traveled by the ruler for each person. We need to compare the distances fallen by the ruler. And we know that it is a free form, vertical form. So we know that distance is equal to U is 0 because it is starting from rest. So U is equal to half GT square, right? If the time is higher, means distance traveled will be higher. So we need to just take the order of. So they are just mean from the higher maximum value towards minimum value. So maximum time will correspond to the maximum distance. Maximum time out of here is this 0.22. So B is the maximum. After B, which one? E. After E, A. After A, D. And then at the end, C. So this should be the order. B, E, A, D, C. B, fourth one. Okay. Consider two uncharged capacitor of equal capacitance, 200 picofarad. One of them is charged by 100 volt and disconnected. Now the capacitor is connected to the uncharged capacitor, the amount of electrostatic energy. So once you charge it, the first one will gain the charge, get the charge. Let's say that charge is going to be C into V. That let's say C1 into V. And then you are connecting this to the uncharged one. This one is charged. You are connecting. So charge distribution is going to happen, right? Unless the both comes on the both gets the same potential difference, C1 and C2. Let's say this is C1 and this is C2. In this process, energy loss formula is, finally we get that energy loss is half C1 C2 upon C1 plus C2 into potential difference across initially across the first capacitor minus potential difference across the second one whole square, right? So you can put all these values to get the answer. C1 value is given to be 200 picofarad. 200 pico means 10 power minus 12. And equal capacitance. Both are equal capacitance, means both are C1 and C2. I can say whole square. And divided by, if we add them, it will become 400 into 10 power minus 12. And into Initially the V1 was 100 volt and it is connected to V2 0. So 100 minus 0. Right? If you are going to solve it, simplify it, it will come 0.5 microjoule. 0.5 into 10 power. So this is the answer. This 11th standard while conducting an experiment to determine the effective length of simple pendulum of length L. Data time taken complete 30 oscillation. The pendulum is completed in 60 seconds. So what is the time period of the pendulum? Time period of the pendulum is time of one oscillation is called time period. Time period 60 seconds divided by 30, which is going to be 2 seconds. And then they are asking length of the pendulum. Given that pi square is equal to 9.8 and g is also equal to 9. So by that way also you can get the idea. This is the second pendulum, okay, and the length of the second pendulum is approximately 1 meter, right? You can calculate also. T is equal to 2 pi root over L by g. Length we need to calculate, so we know that T square is equal to 4 pi square L by g. L is equal to 4 pi square and g will cancel. If we do T square by 4, which is T is 2 square by 4, which is going to be 1 meter. So answer is 1 meter. All right. The peak value of alternating current is 5 ampere. We know that alternating current goes like this, right? With time. This is the peak value. This peak value is given to be 5 ampere. And the frequency is given to be 60 hertz. So from frequency we can calculate the time period. Time period is going to be 1 by 60 second. How long will the current starting from 0? Let's say this is starting from 0. How long it will take? How much time it will take to reach the peak value? So this time they are looking for. We know that from here to here time is called one time period, right? So time is from here to here is this is T by 2 time, and half of that this is T by 4 time. Right? So from 0 to peak value, the time is taken by T by 4, which is 1 by 60 divided by 4, 1 by 240 seconds. This is the answer, right? In interference and diffraction, the light energy is redistributed. It reduces in one region, producing a dark film. It increases in another region, producing a bright film. As there is no gain, there is loss. These phenomena are consistent with the principle of conservation of energy. Okay. So, look, here is the statement 1. As there is no gain or loss of energy, these phenomena are consistent with the principle of conservation of energy. So that's true, right? Diffraction and interference are characteristic exhibited only by light waves. No, that is not true. All the waves shows interference and diffraction. Like if you go for the sound wave also, if you change the path length, you are going to see the different intensity. And you also know that the sound can bend through the doors also. So that is diffraction. So this is false, but this is true. So A is true, but B is false. Okay. A box of mass 15 kg is kept on the floor of a stationary trolley. Let's say this is the trolley. And here is the box kept. Okay. 15 kg mass is there. The coefficient of static friction between the box and the trolley is 0.12. The mu value is given to be 0.12. keeping the box in stationary state over the trolley, the maximum acceleration with which the trolley can be moved horizontally. At what acceleration we can move it horizontally? Ensuring that if an observer is standing here, he is seeing it at stationary. There is no relative motion. So you know there is a limiting friction concept. So if you look at the free body diagram, to match the acceleration, friction will be keep on increasing until the maximum value. So acceleration without any relative motion, acceleration is going to be happen Fs max divided by the. Since Fs max is equal to mu times n is equal to mg only in this particular case. So it's going to be mu g. A is going to be equal to mu times g. Mu is 0.12 and g is given to be 10 here. So acceleration is going to be 1.2 meter per second square. Option 4 is correct. The sum of the kinetic energy and potential energy of simple pendulum 0.02. The speed of simple pendulum bob at the equilibrium position. We know that at equilibrium position, the speed of the bob is maximum, and that is the position when all the potential energy will be converted into kinetic energy, and that kinetic energy will be equal to the total energy. So here, this is given the total energy 0.02. Total energy of this is 0.020. And we know that this must be equal to the maximum kinetic energy. This is Vm is the maximum speed. So we need to get this speed of the pendulum, Vm value. We can calculate it. The mass is given to be 20 grams. We can use that. Convert into kilogram. And you can solve it. Right? I think you are going to get after solving 1.141 meter per second. Okay? But this is the way we can solve. 4 statements are given. A is the mass number here. The first statement is the volume of a nucleus is proportional to A to the power 1 by 3. No, radius is proportional to A to the power 3. And since volume is proportional to R cube, so I can say volume is proportional to A. B option. The volume of the nucleus is proportional to A. Just now we are observing volume is proportional to A. So that is the correct statement. So that is false. That is true. See, the difference in mass of an atom and its nucleus is called the mass effect. No, the difference in mass of its constituent of the nucleus to the mass of the nucleus is called mass effect. It's not just you need not, it's not that you just remove the mass of electron. That is not. So this statement is false. This is not true. False. Okay. And the difference in mass of a nucleus and its constituent is this is true. Then, which are the statements we are finding true? There are 4 statements, right? So B is true and D is true, and rest all are false. So B and D are true, but A and C are false. This is the correct one. The angular speed of a flywheel is increased from 600 rpm to 1200 rpm in 10 seconds. The number of evolutions completed by the flywheel during this time. Okay, so you are increasing the speed of the flywheel, and in such cases, we consider it is increased with the uniform rate. Means acceleration, angular acceleration is uniform. In such case, what we can do, we can calculate theta in radian. And since it is a uniformly accelerated motion, angular motion, we can directly use this formula, omega 1 plus omega 2 divided by 2 into 9, right? omega 1 is equal to 2 pi n1. Omega 1 is equal to 2 pi 600. If you convert into, this is in per minute, you convert into per second. So this kind of thing, and omega 2 is equal to how much? Time is given to be 10 seconds. We can put all these values to get time. Right? So it's going to be this is going to be 25. This is going to be 45. So here 20 plus 40, 65 divided by 2, which is 35. Time is 10. So it is going to be 300 pi radian. So this much angle flywheel has rotated. So how many revolution it has made? In one revolution, it makes 2 pi radian, right? So number of revolution n is going to be number of revolution is equal to theta divided by 2 pi. One. So answer is 1. A submarine is designed to withstand absolute pressure of 100 atm. How absolute pressure? Mind it, it is absolute pressure, not the gauge pressure. How deep can it go below the water? You know, if this is the sea water, okay. If I want to calculate, let's say this is the ship here at height h below. Here the pressure is atmospheric pressure. And if the rho is the density of the liquid, then pressure, absolute pressure at this point is going to be, on the ship is going to be P atmospheric plus H rho. What they are asking? How deep means they are asking H. So H can be written as absolute pressure that it can withstand minus atm divided by density. And you can solve this. This is 100 atm minus 1 atm and divided by density is given to be 1000. G value is 10. Now convert this atm into the Newton per meter square because you are putting everything in SI unit. So 99 into 10 to the power minus should be, I think it's wrongly written, plus 5. Okay. And divided by 1000. So it's going to be 99. This, and here it should be plus 5. 1 into 10 power 5. Now again we need to match the list 1, list 2. Microwave is connected with the generation in production happens by this magnetron wall method. Visible light comes under electrons when it moves from higher level to lower. Gamma rays is the result, it's produced by radioactive decay, and infrared it is the vibration of atoms and molecules. Okay, so you can match it. Which of the following statements are correct? Inside a conductor, the electrostatic field is 0. That's true. Electric field at the surface of the charged conductor does not depend on its surface charge density. No, it depends. If it is sigma, if any conductor is there, sigma is the surface charge density. So immediately electric field is going to be equal to sigma upon epsilon. So it depends on the charge density. So this statement is false. So this is false. The interior of charged conductor can have no excess charges in static situation. That is also true. At the surface of charge conductor, the electrostatic field must be normal to surface at every point. That also we know that is true because it is a equipotential surface, right? So this is also true. The electrostatic potential is zero everywhere inside the charge conductor. No, it is equal to the potential at the surface. So this is false. So we need to figure out the correct one only. Which are the correct one? A, C, and D. A, C, and D. Okay. The work function of the metal is given. Which of the following wavelength of the incident radiation does not give rise to the photoelectric effect? Okay. So let's calculate the threshold wavelength first. So we know that threshold wavelength is equal to work function is equal to, we know that Hc by lambda naught. So lambda naught is equal to, if you are going in electron volt, this will come in angstrom. If you are putting is 12,400 divided by 6.6. Okay. Now this comes around, this lambda naught in nanometer, if you calculate, it comes around 187.7 nanometer after calculation. If we put it 10 down, it will come in nanometer. So it will come like this. So for photoelectric, we want what is the condition? Which of the following wavelength of the incident does not give rise to the photoelectric? We don't want photoelectric. Okay. So lambda should be less than lambda naught. So it should be less than this. Okay. So less than value will be giving the photoelectric. We don't want. So in that case, it will be 200 lambda greater than lambda naught will not allow the any. But this is for the photoelectric effect to happen. Photoelectric effect to happen. This is the condition, right? In the first excited state of hydrogen atom, the energy of its electron is this much. The radial distance of the electron from the hydrogen nucleus in that case is approximately. Basically, they are looking for the radius of that orbit. We know that first calculate the quantum number. So this is equal to minus 13.6 upon n square. If we compare, it is anyhow it is given that first excited state means n is equal to 2. So basically, we need to calculate the orbital radius of n is equal to 2. You can use simply that equation also. If it is a hydrogen atom, directly we can use that 0.529 into n square angstrom. So that will come around 2.1 into this much angstrom means in meter. If I convert this meter, 2.1 into 10 power minus 4. When forward bias voltage statements are given, when forward bias voltage across a PN junction diode increases above a certain threshold voltage, the diode current increases significantly. You remember the VI characteristic in forward bias? Initially, it will be almost nil. Suddenly, it will be increasing after a certain when the barrier potential breaks and all. We remember that. Okay. So that's true statement. Statement is true here. Now, this current is called reverse saturation current. In forward bias, we don't define anywhere there is any saturation at all. It happens in the reverse bias rather. Situation happens in the reverse bias. This we take in microampere. Here we take in milliampere, right? So saturation occurs in the. So it has nothing to do with the forward bias. So this statement is false basically. So I can say statement A is true, but B is false. A flask contains argon and chlorine in the ratio 2:1 by mass. The temperature of measured is mixture is 27 degrees centigrade. The ratio of root mean is square speed of the molecules of the two gases. atomic masses of argon and chlorine are given, right? You know what is the formula for RMS velocity? It is root over TRT by M. It has nothing to do with the mass, mixed mass ratio and all those, this ratio and all those. It has to do with the just molecular weight and temperature, right? Since temperature remains constant for both the, argon also and chlorine also. So I can say that the RMS is inversely proportional to 1 upon root of the molecular mass. So I can say, take this ratio directly from here. It's root of, since it is inverse ratio, the molecular weight of chlorine divided by molecular weight of argon, which is root over chlorine is given 70 and argon is given how much? 40. So it's going to be 27 divided by 2. So answer 1 is correct. Okay. I think we are all set. So hope you understand all these questions. Thank you. Thank you very much.